Okay, we're going to go ahead and get started. Good morning, everyone. I'm Greg Daniel, Deputy Director in the Duke Margolis Center for Health Policy. And I'd like to welcome all of you to our workshop, Safety Assessment for Investigational New Drug Safety Reporting, which is being convened under a cooperative agreement between the FDA and the Duke Margolis Center. We're pleased to be joined for these discussions by a range of leading experts from across the stakeholder community, including government, academia, industry, and others for a productive exchange of the issues at hand. Today's focus of this workshop will be the FDA's draft guidance that was issued in December of 2015 entitled Safety Assessment for IND Safety Reporting. This guidance is the latest in a series of policy developments issued by the FDA to improve the efficiency and quality of IND safety reporting and to help sponsors of INDs to identify and evaluate meaningful safety information that must be reported to the FDA. There were a number of recommendations provided in the guidance which outline a systematic approach for safety assessment and seek to clarify key issues relevant to sponsors' review of aggregate data for IND safety reporting. Examples of the important issues addressed include how sponsors might evaluate unblinded data from ongoing trials and determine thresholds for IND reporting while maintaining trial integrity. Additionally, the 2015 guidance provided information on developing a safety surveillance plan and defining the role and responsibilities of the Safety Assessment Committee. Comments received thus far to the FDA on the draft guidance highlighted some potential implementation challenges with these issues, and today's goal is to hear from a range of stakeholder perspectives on and experience with implementing the draft guidance to better understand the approaches being used, the key challenges faced, and to identify areas and potential approaches that could inform the final guidance. We're looking forward to having a robust discussion on these issues and hope you will contribute to the discussion throughout the day. Before we get started, I'd like to quickly go over the agenda. We're going to have a series of presentations that will frame and tee up key issues for discussion. First, we'll hear from senior leaders at the FDA. Bob Temple and Peter Stein will provide a background on IND safety reporting, an overview of the recommendations in the guidance, and challenges that they've heard from sponsors with implementation. We'll then turn to Janet Witz and Robert Baker, who will be presenting key stakeholder views from outside of the agency and raise important considerations for implementing the draft guidance recommendations. After these presentations, we'll spend the rest of the day taking a deeper dive into several of the major issues covered in the guidance. In session one, we'll discuss and focus on challenges and best practices for designating anticipated adverse events and systematic approaches for predicting the rates of these events. Following that session, we'll take our first break at around 10:50 and then reconvene at 11 for session two. And in that session, we'll address two separate but closely related issues that are related to unblinding of data for safety assessment analyses and whether trial integrity can be maintained with periodic unblinding by a group other than the Data Monitoring Committee. The panel will then consider, related to that, intended functions of the safety assessment committees and whether such functions could be addressed through other bodies such as the DMCs. We'll then break for lunch at 12:30 and then reconvene for session three at 1:30 to discuss key issues pertaining to when events in clinical trials may indicate more frequent occurrence in the treatment group compared to the control and the issues and challenges related to that. And then we'll take a quick break at 2:45 before our last session of the day, which will tackle key data and methods issues associated with conducting aggregate analyses of patient-level data pooled across study arms on multiple studies. Just a few housekeeping notes before we kick off with the opening presentations. As you'll note in the agenda, each session will begin with a presentation or two, followed by reactor comments, and then moderated panel discussion. We'll also have time in each session to set aside for broader discussion with the audience, and for those in attendance, we have roving mics for you to use throughout the day. Given the storms impacting the East Coast over the last couple of days, at least one or two speakers will be calling in during the panel discussions. I want to remind everyone that this is a public meeting. The event is being live webcast, and so everything you say will be part of the record. And for those of you joining us through the webcast audience, we encourage you to participate in the day's discussion by sending questions that you have to a special email address, and here it is, margolis.ind@duke.edu. And it's not written out there, so margolis.ind@duke.edu, and our staff will forward those questions to the moderator. For those of you in the room, feel free to help yourself to coffee and beverages throughout the day. And then lastly, during the lunch break, lunch will be on your own. There are a number of restaurants close by, and we'll be starting promptly at 1:30. So if you're not able to finish, feel free to bring anything that you have, your food, back into the room here. Finally, just as a reminder, although this meeting is being convened under a cooperative agreement with the FDA, this is not a federal advisory committee. We won't be trying to reach consensus. We won't be taking a vote. And this meeting will be successful if there's an exchange of lots of ideas and open discussion. So let's go ahead and get started. I'd like to turn this over to our FDA colleagues who are here to help kick us off. First, Bob Temple, Deputy Center Director for Clinical Science, Center for Drug Evaluation and Research at the FDA. Bob will give a brief presentation on the regulatory history of IND safety reporting. After Bob, Peter Stein, Deputy Director of the Office of New Drugs, Center for Drug Evaluation and Research at FDA, who will discuss some of the challenges that he's and FDA has heard from sponsors faced with implementing the 2015 draft guidance. Thank you. Bob?
Okay, good morning. Who would have guessed this would all be so hard? Okay. No? I push the arrow. Okay, so that's what this is about. All right, that's a little bit of the history. We first put out a proposed rule in 2003, then we got the final rule in 2010, and guidance in 2012, and that's last one is not 2005. It's not 205. It's 2015. And it's worth remembering why we did this. There's a perfectly obvious requirement to report important adverse events under the IND so you can monitor patient safety. And the way the regulation was written, it just said if there's anything serious associated with the use of the drug, you should report it, and that meant a reasonable possibility, but it wasn't defined any further. So what we were getting is in a cardiovascular outcome trial, we'd get every serious event that happens, and these people would have heart attacks, strokes, and all those kinds of things, and they'd all be reported. And one, it was a lot of work for us and not very helpful because those weren't mostly related. But it also sort of kept people from looking properly. You weren't really diving into the data to see what it meant. So before the rule in 2010, the events reported had to be associated with the use of the drug, and that meant there was a reasonable possibility. We tried to eliminate events that were probable manifestations of the underlying disease or that were adverse events common in the study population, even if they weren't exposed to the drug, things that just happen in people of that age, heart attacks, strokes in older people, and so on. And people were also reporting study endpoints, so we didn't want any of that. So in 2010, we introduced the term suspected adverse reaction that made it clear there had to be a reasonable possibility, which also meant that there was some evidence to suggest a causal relationship, not just that they were taken together, not that the event happened in someone who took it, but that there was reason to believe it might be related. And then, as usual, you had to report within 7 to 15 days any of these things that turned out. And the rule identified 3 different kinds of things, 2 of which are easy and not a problem, the third of which is why we're here. Uncommon events that are strongly associated with drug exposure, Stevens-Johnson, agranulocytosis, things like that, those are usually drug-related when they happen. It's pretty easy. then there's some odd things that aren't commonly associated with drug exposure, but really don't happen in the population. And the example we gave was tendon rupture with certain drugs. But the final one, actually that says 3 easy and 1 hard. That's wrong. It's 2 easy and 1 hard. The first 2 I did were the easy ones. And then the final ones is doing an aggregate analysis of an unexpected adverse event that occurs more frequently in drug treatment group than in the control. Unexpected, by the way, means it's not in the investigator's brochure. That's all unexpected means. We've from the beginning tried to get people to understand what kinds of unexpected events could be anticipated in the population. We talk about that a lot. So the 2 things that need to be looked at with an aggregate analysis are events that come more frequently in the drug treatment group, serious events, and things that we already suspect, because they're in the investigator's brochure, but that are occurring more commonly than we thought they were going to. We haven't had a lot of discussion of that. So in a 2012 guidance, we tried to clarify what all this meant, and we asked for a periodic review of the accumulating safety data and appropriate reporting. Obviously, to protect trial integrity, sponsors need a predefined safety monitoring plan with processes and procedures for review of the safety data, including the frequency of review, and of course, they also have to define how they're going to keep people from knowing results they shouldn't know about. And the guidance addresses the role of unblinding in determining whether a single occurrence of an event needs to be reported. Obviously, you have to know whether, if there's agran, you have to know whether the person was on drug or not. And then as part of the aggregate analysis, if you just report the events, it's of no use to anybody. You really have to unblind it to make sure anybody knows what's happened. We wrote a further guidance in 2015 to try to clarify all the issues that had arisen, and they're not easy. What's the reporting threshold? I mean, if you're looking at some event that's expected in the population, heart attacks or something like that, how do you know when to unblind and look? And then when should you report? If it's 20 versus 18, how do you decide it? And that's a very hard question. I know we're going to talk about it. So we had a meeting in 2015, and we learned that most people were not really doing what we had hoped they were doing. They were still reporting all of these events. A couple companies, I don't think I'll name them, had been looking at the data properly. Lilly had published work on this long ago and had been doing it for a long time. But we found that most people weren't doing it. So we took a look, actually, at the analysis of serious adverse events submitted in oncology trials, and they just mostly weren't informative. They were reporting everything that happened, not really distinguishing. So thoughtful discrimination of adverse events that are drug-related from those that are not was generally not occurring. Most people were reporting just what they'd always done, a sort of data dump. The guidance was intended to enable sponsors to evaluate unblinded data from ongoing trials when that was reasonable, to determine if the threshold for IND reporting, not well defined, while maintaining trial integrity. So they needed a safety surveillance plan. They should say what they were going to do so everybody would know. And we recommended, we've taken the official letters away, but a safety assessment committee, a group whose obligation was to look at the unblinded data, a very limited group. We didn't want to damage the trial or anything like that. We also suggested that any analysis like this should look at data from all ongoing trials, not just one by one, and factors to consider in the reporting threshold. But it's hard to write a rule on that. Okay, I'm done. That's what we were trying to do. And now here comes Peter.
Thanks, and good morning. Appreciate everyone weathering the storm, particularly those who came from the Northeast. What I'm going to try to do is to sort of follow on Bob's comments, who I think laid out a lot of the issues, and see, talk just very briefly about some of the particular aspects of the 2015 guidance. I'm going to mostly focus on the feedback we've received. What have we heard from sponsors and from other individuals about some of the challenges in implementation of the guidance, and some thoughts and strategies that we've been hearing about regarding how these issues in the guidance can be best addressed. So let me start by just touching on some of the particular aspects of the guidance. I think most folks here are quite familiar with it, and Bob has touched on this. The 2015 guidance expanded on the safety surveillance plan. It provided a greater definition of the roles and responsibilities of the Safety Assessment Committee of the SAC. It talked about membership. It talked about the data that the SAC would review. It provided a clear recommendation for unblinded analysis, for regular periodic unblinded analysis of aggregated data pooled across the program that the SAC would review. And this was focused, as Bob mentioned, on those events that really only could be identified as drug-related by looking at aggregate analysis and by looking at unblinded aggregate analysis. Although the guidance did talk about an alternate approach, which was to look at the incidence of events in the blinded aggregate database and look for whether it surpassed a trigger or threshold value. It did have, I think, a useful discussion on factors to consider and how one determined whether there was an association with drug or not, although this is obviously a complicated issue which requires judgment, and there's certainly going to be uncertainty there. And then it did talk about the importance of maintaining the blind of the need for a firewall and ensuring that was appropriately implemented. So I think Bob's already talked about this, and we'll be thinking about this through the day. There's clearly several different sort of categories, if you will, of the types of IND safety reports. The first two categories, those that are typically drug-associated types of events, straightforward. That can be obviously seen in blinded analysis. If you see a Stevens-Johnson, clearly there's a substantial concern that it may be related to the drug. The other category are those events that can occur in a background population, but are unusual for this population in the study. So, for example, myocardial infarctions occurring in a quite young population would be an event that would be potentially seen in other populations, but would raise a concern about drug relationship in a young population. But really what we're talking about are those events that are part of the background related to the concomitant medications, perhaps related to the underlying disease, things that can occur simply related to the background population and its comorbidities. And these will be seen in the background with the trial. What we're really looking for is when there is a meaningful imbalance in those events, and that could really only be detected potentially by looking at unblinded analysis in aggregate. And the challenge there is how do we do that, and how do we identify those events for appropriate reporting so we can assure that patients are informed and appropriate decisions are made about the trial. So what have we heard? We've been getting feedback on the 2015 guidance, and what I've tried to do is to categorize the feedback into a couple of different categories of types of points that have been made. So I'll just give you the highlights of the categories, and then I'll talk a little bit more about each of these categories and what the specific challenges have been. So first, trial integrity, the concern around disclosure of unblinded data related to the unblinding and expanded numbers of individuals who will see the unblinded information. The greater trial complexity, we have a new infrastructure with the SAC, now also supporting the DMC, the SAC, the sponsors' internal committees, and so the complexity of the trial being increased, resource requirements, and also the overlapping responsibilities there. Separating signal from noise. This is clearly a key issue. How do we determine where there is a meaningful imbalance? When we're looking at multiple comparisons, we're looking at multiple analyses, we have ongoing trials. How do we pool and how do we look at imbalances in a way to make sure that we're identifying important imbalances but not reporting false positives? And then there were comments about what is the need for this act? Can a DMC role be expanded to provide appropriate reporting where there is an important imbalance? So let me drill down to these a little bit more in the next few minutes. So with regard to trial integrity, we've heard about the concerns regarding repeated unblinding of the interim data now for the SAC, and also as previously for the DMC. There's more internal sponsor personnel potentially involved, whether directly participating in the SAC or in supporting the SAC. Obviously, this makes the assurance of a firewall a little bit more challenging. There's an impact, of course, if there's more IND safety reporting and unblinded reports of more common events. Could that impact trial integrity because of changes in behavior of investigators and management of patients? Challenges related to trial complexity and overlapping responsibilities. This is a new committee. It has to be folded in to the ongoing program infrastructure of the DMC. There's the internal sponsors safety committees. The requirements for supporting that, the challenges of setting this up and making sure it's appropriately integrating. And we saw a number of comments about the complexity of the relationship. How do we assure appropriate responsibilities allocated to the SAC versus the DMC and the internal safety committee from the sponsor? How do they communicate? How is it clear who's responsible for what? Are there potential for breakdown because of the greater complexity of the numbers of committees and the numbers of individuals responsible for assuring safety of the patients participating? And then there were quite a few comments about the issues around separating signal from noise, the challenges of pooling. There were a number of comments about that we're dealing with a program that's ongoing. There are trials at different stages. They may be for different populations, different indications, different doses, different regimens, designed differently. Randomization ratios may not be the same. How do we pool completed and ongoing trials, different designs in different populations in a way that is meaningful and accounts for these differences and avoids getting false positive imbalances related simply to these complexities of the program? And when do we determine, how could we determine when an imbalance should be reported? We're looking at a challenge of multiplicity here. We have multiple different comparisons because there'll be multiple different adverse events. We have multiple different looks at those. How do we assure that when we see an imbalance, it's really meaningful? Do we report, as Bob said, 20 versus 18, 20 versus 2? What is our threshold for determining that there really is an association? Because in any large trial database, there will be imbalances in one and the other direction that may not be meaningful. How do we decide which of those are meaningful, are important to report so that we can make patients aware and appropriately implement changes to the trial if needed to assure patient safety? We did hear comments about the alternate approach, the idea that we would set up a threshold for different adverse events and only report when the aggregate blinded data exceeded that threshold. Okay, well, how do you decide what that threshold should be? Where do you get the data from that says this incidence is the threshold? Above that, we should unblind that particular event and report it. And I think we'll hear some creative solutions today to that particular challenge. We also heard about the expertise of the SAC. Do they really understand the drug and know what the likely adverse events might be that might be seen related to the drug or in the background population? How do we get the right experts looking at this data in the most informed way? And finally, there were comments about whether the SAC was needed. Could this be managed by the internal safety committee from the company in concert with the DMC? Would that mean a change in the role for the DMC? How would that be done? Could they manage to look at unblinded reports? So that was also something that was raised. So the goals for the meeting today, as I think you've already heard, but I'd like to just reemphasize, is really to understand input from all of you about the challenges you're seeing. Are these the challenges? Are there other challenges? And in particular, what are the solutions that you've come to? How have you addressed these concerns? We'd really like to hear your innovative, creative ideas about how these challenges can best be met. And obviously, our intent here is to identify the issues and the approaches that can inform as we think about moving from the draft guidance into the final guidance. So I'm looking forward to a very interactive discussion today. We certainly appreciate your weathering the storm if you've come from the Northeast, and appreciate your attention and your input. So thank you very much. We'll move on.
Great, thanks, Bob and Peter. So next, we're going to hear from two perspectives outside of the FDA on some of their experiences with these challenges. Robert Baker is Vice President of Global Medical Affairs at Eli Lilly, who will give a brief presentation based on the work being completed by the Transcelerate Biopharmaceutical member organizations regarding their perspectives on the FDA's IND safety reporting guidance. Following Robert, we'll hear from Janet Wittes, president of Statistics Collaborative, who will discuss key challenges from her perspective with the IND safety assessment and reporting. So I'll go ahead and turn it over to Robert.
Good morning. Thanks to the members for coming. Thank you for Duke Margolis for inviting us to present on behalf of Transcelerate. Greg, the title was a little bit out of date for me. I formerly was in Med Affairs, but now have three groups I'm responsible for at Lilly, and the salient one is our global patient safety group. But I'm speaking today not on behalf of Lilly, but Transcelerate, where I've been the initiative leader on a work stream that we're going to describe for you called Interpretation of Pharmacovigilance Regulations. And I'd be remiss to not thank the agency for their interest in gathering perspectives. You'll find this perspective mostly confirms what Drs. Temple and Stein have already laid out for us. First, a little bit on what Transcelerate is for those who aren't familiar. Transcelerate's been in existence since 2012. It has a small central group, but it's funded, and the work is mostly done by member companies in the interest, ultimately, of improving our effectiveness in developing new medicines, but certainly with an emphasis that's relevant here as well, on not just effectiveness, but productivity for unit of effort that goes into it. Since 2012, it's actually been a fairly successful experiment. I know that from the clinical development side of my work, where many of the initiatives used by Transcelerate have led to us being more productive together, or helping each other together in a pre-competitive sort of way. Transcelerate has grown over time, and if on the far right is now up to 19 member companies, with Novartis joining most recently. And these are mostly small and mid-sized companies captured in the tiny print up above, so a lot of American companies, but also European and Japanese. And together, although it's a minority, of course, of the companies within the biopharmaceutical industry, it accounts for about 70% of the volume by revenue across the industry. I'd highlight in 2016, and my numbers are doing the same as others that we saw today. They're dropping off the line, but BioCelerate was launched. That's of interest to this. It's actually not in the clinical space, it's preclinical space, but we're quite excited within the safety group about the potential for the sharing of toxicology data, hopefully advancing our ability to, if you will, seek safety by design and try to conduct more of our safety work before it's pharmacovigilance and in the clinic. And as well, in 2017, although not focused on safety, one of the trying to find it on here, maybe dropped off from the last version that I saw, but working on sharing of placebo data across disease states in which many companies are working in order to help us try to create a more robust understanding of what background is that we might expect, which of course could be relevant to today's conversation. Turning to the present discussion, I was taken a little bit aback, Greg, when you mentioned that there's experts speaking today, because I wouldn't count myself in this, but I led a team of experts, and I'd point out that for our team, Sadika Mian from Amgen was the leader of the group working on the IND reporting question, as well as Rachel Mercado, who's here from PwC, who was our capable project manager through this. In 2017, should have mentioned on the last slide, Transcelerate decided to build on what we took as member companies as success in the clinical space, to look at other areas in which companies were engaged, where the methodology of pre-competitive sharing of ideas and solutions would help. And of many that they considered, pharmacovigilance was the one that was chosen. And the work that we did last year is actually, I would think of it as a demonstration project regarding the question of whether the Transcelerate methodology would be similarly effective in advancing what we do. We found, for example, this project on IND reporting much more work than we expected, but we also decided that what we learned took a lot of work that was less than we would have done pursuing it cumulatively individually, and that was a bit of the intent with this. So we talked about ambiguous regulation. We, of course, from around the globe get frequent changes in regulation, and occasionally there's ambiguity in the intent. More often the ambiguity is in how to translate that intent into actually operationalizing it within our companies. And the intent or the value in this is actually hopefully one that's very tightly shared, which is effectively understanding safety. And we thought that the efficiency for us would also be a gain in quality, because not every company has a depth of expertise in every aspect. And so by sharing together, we could more adequately obtain that. We found that was the case. I hadn't thought about it, but we also found that not every company has the same visibility to thinking or nuance or clarification that we get individually in conversations with regulators, and that was quite useful in sharing together. And that through that, we, in terms of the benefit to us, would be more likely to misinterpret and therefore become outliers, putting ourselves at inspection risk. And finally, that as I think Duke Margolis is helping us with today, that perhaps together we could focus better on topics that are best raised or bubbled up for discussion with those who do set our rules. This group, I was a bit surprised. I thought that we would want to get a newly breaking bit of regulation to work together on. But as our member companies got together and started working, they highlighted the 2015 draft guidance on IND reporting as one where there was a lot of enthusiasm for taking it on, which I think you can take as a marker that companies individually were finding it difficult, or were finding a bit of anxiety about whether we'd quite gotten it right as the agency had intended. This rehashes what you've heard already, so going on to our method, which was fairly simple, but as I said, took a lot of work. Sadika and the team from, I think, about 5 or 6 other companies got together and first went line by line through the guidance, and found probably would be fun someday to show you a highlighted version, because it was embarrassing how many things that we had highlighted that people weren't quite sure whether they'd gotten right, what to do about it. But then based on that, compiled a survey, a lengthy survey, that we sent to experts at each of the individual member companies to try to get feedback on how we had interpreted, how we had responded, how we're operationalizing against this. They found a number of follow-up questions, and we've completed results, and now we're at the stage of beginning to share them, and of course have available much more detail behind what I'll report now, which actually is just this slide. I'm going to put, these are our conclusions. I'm going to have a couple of slides to go into more detail on the second and third bullet point. The first was the companies had existing approaches and have adapted those approaches that I guess you'd think of as the surveillance plan or the surveillance team for reviewing, accumulating data with the focus on subject protection here. Our expectation is that for patient protection, before approval, we'll have a more comprehensive unblinding and pooling that we would at the point of trying to get emerging events for the sake of the subjects who are in our studies. Where we began to find variability was on the point of how, what were the methods for reviewing the data, unblinding the data, thresholds for reporting, as you heard from Dr. Stein this morning. And the anticipated serious adverse event concept has been implemented not by all and somewhat variably, and often, as I'll get to in a second, with a ad hoc aspect at the level of medical review at the final point. We have, however, seen across the groups that participated average of over 50% reduction in the number of reports we were sending. In the case of Lilly, our estimate is 80% reduction. However, we still have been admonished by at least one division that we are sending too many still amongst that residual. And the last part, all striving to meet the spirit, I'll come back to that at the very end, but I think there was actually, speaking for myself, but I think among most of us, a pretty strong sympathy for what's trying to be achieved through these rules. To dive into the two that were most challenging, this one is on anticipated serious adverse events and documentation within the protocol. That's possible. It's being done by most. It becomes more difficult the more comprehensive one tries to be in doing this. So for example, in oncology space, we typically would have a fairly robust thinking about hematological impacts for a lot of drugs, but there are many, many other impacts that are in the background within cancer that we may not capture there. In terms of background rates, there are a number of approaches for this. I would say that no matter how we get at it, it's perhaps harder than it looks on the surface, because even if we had a very strong ability to estimate over an overall population, clinical trial populations differ in features that are important. Geographies, really subject of who comes into the study, is always going to create some variability in it. Most companies do maintain anticipated serious event in both the clinical database and the safety database. And finally, this last point, that most are managing anticipated events with a medical review as part of a causality assessment. Let me say more specifically what that means in the case of Lilly, because I guess it's a little vague as written here. So we will capture some of this at the front in our plan. However, we also have medical reviewers who will look at each case that might otherwise mail for the confound related to disease state background or other concomitant treatment that would make that individual event uninterpretable on its face. The event that's not itself convincing will be one that we then end up screening out. So this is perhaps not as intended, upfront thinking and documentation a priori, as opposed to medical judgment on the back end, I think reflecting how very broad are the possible events that could happen in the course of a study. And on this one, the aggregate analysis of safety data, this of course will speak both to how this is done and also thresholds, and we have found this to be a significant area of continuing question, so thankfully we'll discuss further today. Approaches differ, such as comparison of rates across treatment arm, pooling across studies as appropriate, comparison of incidence to baseline frequency. I've mentioned already some of the challenges with that. At Lilly, we tend to think of also aggregate being at the level of the product rather than at the level of the trial. I think that'll come up as we talk about DMCs today. When running blinded studies, most companies perform aggregate analysis. However, unblinding is done for cause, and that would certainly be the case for Lilly. I can represent that as well, and a couple of comments that might be of interest to it. So our approach is that the blinded team looks at the accruing events with the expectation that they would adjust as though any event is occurring only in the experimental arm. And therefore, in excess of that, that of course gets more complicated if there's more than two arms, because it's not as simple as doubling, or dose effects wouldn't be easy to portray in this. And it's a judgment regarding whether it's more than the background. I've been surprised. I think across most of our programs it's unusual for unblinding, maybe one or two per program. We had one approval recently in the oncology space, which would have an example where this failed. We found a significant imbalance that's now labeled, and it was an event that is quite often associated with cancer, and yet there was an imbalance that we had not picked up in the course of that of this approach. And so before next steps, let me get to a conclusion. I've mentioned two things I haven't that are sort of general points that are worth thinking about. One is about specificity and one is about harmonization. So one of the interesting things about the draft guidance that those working closely on it would understand even better than me is that in some cases they thought it was more helpful than necessary, in that there's a lot of differences that are hard to predict on the front end between a given drug, a given disease state, or a given company approach, where specificity could actually make it harder to implement against this, or granularity of the instruction. On the other hand, I think the whole reason we got together is for the other direction, where if we don't have guidance about how best to do it. Regarding harmonization, I think you could think of it at different levels. So as most are aware, outside U.S., there's a movement in a different direction, actually sort of pushing companies for more reporting. To some degree, that harmonization should be just our problem, and it is solvable just with more resources in the way that I've mentioned. We have set to meet the U.S. intent, a separate process, manualized process to pull out events. Where it becomes a more common problem is when we get into one of the issues that we've teed up today, which is things like trial integrity, where the harmonization issue either prevents something that's mandated by a different regulatory group, or maybe even more importantly, would collide with another tenet that we are trying to achieve together. But in terms of our conclusion, could not be much more aligned with the 4 topics that you have on today, so we look forward to that discussion. We also look forward, hopefully, to even deeper discussion and work with the agency going forward. Thank you. That was in case I forgot to say thank you.
Yeah. So I'm Janet Wittes. I'm very glad to be here. Thanks to everybody for coming. I feel bad for those up north with the snow. I'd like to talk. Oh, what do I do? That's me. I changed the word from key to some because I don't know what key means in this context. But let me tell you a little bit about my role, I think maybe why I'm here, and then proceed with some of the issues, many of which will be just echoes of what you've just heard, but echoes from a different chamber. So a lot of what I do is sit on DMCs or report to DMCs. And my experience there, after the 2012 guidance draft came out, I would routinely say, either when I sat on a committee or when I reported, that we really should look at the guidance, or I would tell the companies who were involved, hey, you really should come and tell people what the guidance was, and they'd roll their eyes and say, it's irrelevant to you. So I've given up. So that's one piece. The other piece is that I do a lot of consulting with what I consider small biotechs. You've described small to me. Dr. Baker, I don't see you anymore. There you are. What you described as small to me is huge. These are, I'm talking about tiny little companies. And when I bring up the guidance to them, which I do, they not only roll their eyes, they say, no, 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 we're not going to touch those. We're reporting everything because our heads will be chopped off if we don't report everything. So that's where I come from. You all know what the old problem was. Many reports were sent to many people, to the IRBs, to the investigators, to the DMC members, piles of them, and to the FDA, of course. Few were read. Who could read them? There's this great big pile of stuff. And even if you could read them, they were uninterpretable because they didn't tell you who was on what. So the goal, I look at the goal of this set of guidances and movement toward what we would hope is harmonization, is to separate the wheat from the chaff. And that's a complicated thing. How do you send fewer reports but don't miss signals? Sponsors need to assess risk, not versus benefit, and I'll get to why I say that. That's from my DMC hat. Know what to report, and their ultimate goal is to quantify harms. So what are the challenges that this has raised? And you've heard them already from the previous three. First of all, fitting an SAC, a Safety Assessment Committee, into an organization that's already there. You've heard that from Dr. Baker. I think it's a bigger problem than people may realize. It's easy if you start something, to start a new little company, to figure out how to do that. But to integrate a new system into something that has been working and is well-oiled, and now you plop something new, that's a huge issue. I find, as a statistician, that the guidance doesn't really have much of a hint of statistical thinking, no way of figuring out how to how to set these thresholds. There's thresholds to reporting, but how in the world do you set them? There's the issue that Dr. Baker raised about the US FDA versus other countries, and I think that's a huge issue, because if the purpose of this, and I'll get to at the end, is sort of a paper reduction act, and you have to do one thing for the FDA and something completely different for the EMA and other regulatory agencies, that is an increase in burden, not a decrease. And then, of course, as you've heard from all three, who should be blind to what? And that is a huge, I think, a really complicated issue. Who is blind? At what point does the unblinding become so big that it affects the integrity of the trial? So what are we talking about? And I want to go through the words, because those of you who are here probably know what all these words mean. But I venture to say, I will bet money that most of the investigators don't know what these words mean, and a lot of the, and even some of the sponsors don't. So suspected, as Dr. Temple said, there has to be some evidence to support a causal relation. All right, not just my kid took a vaccine and became autistic the next day. You got to have evidence of that. Unexpected, and we're going to get to this word unexpected in a bit, not known to be associated with the drug, or more severe occurrence of the event, or as Dr. Temple said, more common than expected. Serious, distinguished from severe. Now, again, I sit on DMCs, I know a lot of investigators, they don't understand the difference between serious and severe. One is a regulatory definition, and one is, oh my God, my arm hurts. Adverse, I think we all know, adverse means bad. And reaction, as distinguished from the more neutral word event. An reaction is something caused by, that's related to the suspected up at the beginning. And if you look at some of the guidances, those words are sometimes used interchangeably. And that's confusing for people who don't know what the meaning was supposed to have been. Okay, there's this definition. Again, I suspect all of you know it, but you better be telling people that you're working with so they understand. The difference between expected, which belongs to the drug, and anticipated, which is a property of the population. And then we've heard the three kinds of events. The single occurrence known to be strongly associated with drug exposure, and there, I think that's easy, as Dr. Temple said, that's an easy one. It happens, don't agonize, report it. That one's easy. Oh, where's my B? I lost B. Well, anyhow, B is also easy. But it's the C events. It's the events that are, no, that's B. Okay, sorry, that's B. A few, and what does a few mean? You know, there's my status. Is few 1? Is it 2? Is it 3? What is few? Not commonly associated with the drug, so not really expected with that drug, but otherwise uncommon in the population, so unanticipated. So you agonize a little bit, but ultimately report. And I think the importance here, here's this poor little, I don't know whether it's a rat or a mouse, saying, I don't usually volunteer for experiments, but I'm kind of a puzzle freak. It's important to know, because of this word anticipated, what is your population? And then, but the real problem, or the problem of the day, is the type C events. Events that occur more frequently in the treatment group than in the control group. Than one would, more frequently in the treatment group than one would expect. But the important thing is no single case is a surprise. So they may be anticipated events, like strokes and MIs in the elderly. I hate to say that now as I'm getting into that word myself, but infections in kids, those are anticipated. Because if they were truly unanticipated, we dump them into type B. Now, what kind of trials are we talking about with the type C? I think we're talking about randomized, long-term blinded trials. Those are the ones where these unanticipated type C events will occur with a frequency large enough to notice that one group is having a higher rate than the other. And why they have to be randomized, why they have to be blinded, because these are the ones that are problematic. These are the ones we have to make decisions about. How do you know that you have an excess of an event rate? So, question of the day, when should these be reported? And the answer is, well, that's easy. When the frequency is higher in the treatment and control, that's when they should be reported. So when it's 15 days, I mean, we know if you have a Stevens-Johnson, we know what 15 days is. That's from the time that you have the event. Well, what is 15 days for this? It's the time at which somebody recognizes that there's an increase. Is that right? I don't know. To me, that's one of the ambiguities I see. And we have to unblind to know the answer. So there's the rub. Part of the evidence requires knowing if the person took the drug. Because if they didn't take the drug, then the drug couldn't have caused the event. So somehow, all this that seems so simple on the surface is actually not so simple. How do we know? How do we unblind? When do we unblind? And when do we say, aha, there's an excess, we have to report? So, who looks at blinded data in an ongoing clinical trial? Now, this may be very company-specific, what I'm saying. But in general, the participants, they have to know whether they report something. And I think one of the things that really is important to me, or is important, is that we kind of forget that the reporting is coming from them. And if they only know about themselves, right? They're blind, they don't know what they are on. They know about themselves and what the social media is telling them. I had a very interesting case, that's why I went like this. I had Shingrix last week, and I had a huge reaction. I reported to VAERS because I thought, okay, I'm supposed to report things to VAERS. And it's one of the things it says, VAERS is the vaccine adverse event reporting group. It said, it's called severe if you change your daily behavior. So I look at this and I say, well, yeah, I didn't go to the gym because it hurt so much, but not many people my age go to the gym, so I'm going to say no. And I realized as I was doing that the collection of data from the very beginning is something we have to worry about. The investigators, they only know about their participants and what they hear from other people. And the sponsors know the entire development program. So they know a whole lot more, some blinded and some unblinded, but they don't have access to data from other drugs in the same class. Who looks at the unblinded data? Mostly the DMCs and the SACs now. But the DMCs don't think of risk. They think of risk versus benefit. They think the questions that they ask is, are the participants safe? And that means, is the likely benefit outweighing their likely risk? And therefore, they'll be pretty tolerant of adverse events early on when you don't see benefit. And they're asking also, is the trial still asking an important question? They're very reluctant to report out. And they have a structural limit because they only know about their trial, not about everything about the drug, and they certainly don't know about drugs in other classes. What do you guys bring to the table? You bring both blinded and unblinded data. You bring other drugs in the class. And so, when a company gets a question about, would you look at neuropathy? And they scratch their head, we haven't seen neuropathy. Aha, somebody else, some competitor must be having that problem. But not much data from large ongoing trials. Okay, question. How long should participants be followed after they stop study drug? This pertains to how safe things are. the rule of thumb is, well, 5 half-lives, the risk is gone. But that ignores a cascade of events that might occur after people stop taking a study drug, which could underestimate harms. On the other hand, if people drop off of the study because they're feeling sick, and they drop off more in the control group, you can overestimate harms. So failure to follow a long time can under or overestimate harms. So here's a summary. Who sees what? The sponsor is blind to the ongoing trial, full access to other data from trials of the same drug, if that have been finished, and no access to drugs information, ongoing information from other drugs in the same class. The DSMB is unblind to its own, the study it's looking at, but blind to everything else, and the FDA has a different kind of data. The question that I think we're going to be facing today is, how do we marry these three? Are there cultural, operational, and legal and proprietary roadblocks to marrying this? Even if we succeed, will we identify harms more quickly and reliably, which is that's the ultimate goal, or is this just an elaborate paper reduction act? Thank you.
Okay, great presentations from Robert and Janet, and we should have all of you more often because you guys are exactly on time. This is wonderful. So now we're going to go ahead and kick off our first actual session on the agenda, and I'll ask the panelists to come up to the stage, and then I'll turn things over to Jacqueline Corrigan-Curry, the Director of the Office of Medical Policy, Center for Drug Evaluation and Research at FDA, who will be moderating this session, which is about identifying serious anticipated events. So Jacqueline, thank you.
Okay, thank you. I'd just like to call the panelists to the stage, and then I'll do some introductions, and we'll have our presentation and then our discussion. And we've heard a little bit about the challenges with identifying the anticipated events, which is really at the heart of when we're going to decide which events are not going to be reported as single events. And so that's what we will focus on. We'll try and stay in that swim lane. I know it's hard sometimes not to get into other issues about thresholds and things like that. So first I'd like to, why don't you come up, and then I know they're waiting for a mic. I'd like to introduce all our panelists, and then we'll get started. So first I'd like to introduce Barbara Hendrickson. She's in the Immunology Therapeutic Area Head in Pharmacovigilance and Patient Safety at AbbVie. I'd like to introduce Chris Granger, Professor of Medicine and Director of the Cardiac Intensive Care Unit at Duke University Medical Center. Adrian Dana, who's the Vice President of Global Patient Safety and Risk Management at Alnylam. And Mary Ross Southworth, who's our Deputy Director for Safety, Division of Cardiovascular and Renal Products, Office of New Drugs at FDA. And so as soon as we get a mic, we're getting our mics now, Barbara's going to lead off the session with a presentation.
These are going to go live in about 30 seconds.
Okay, maybe I'll get started. The reason I'm presenting today on behalf of AbbVie is because I'm one of the co-leads of our Safety Assessment Committee initiative that's been going on for the last 2 years. And I just was asked to speak about identifying expected adverse reactions and anticipated events. And of note, the presentation contains opinions and positions that are my own and not necessarily of my company, AbbVie. As we've already heard today from previous speakers, anticipated serious adverse events are those that the sponsor can foresee as occurring with some frequency in the study population, independent of investigational drug. And those are not interpretable on the basis of just one single event. And so we can't conclude with a reasonable possibility that the investigational drug caused that event. And these serious adverse events that are anticipated may fall into several different categories, including known consequences of the underlying disease, events that are common in the study population independent of the investigational drug administration, so events that occur in the general population that may be included in the clinical trials, events that are known to occur with the drugs that are being administered as part of the background regimen that might be required for participation in the clinical trial, and then series of adverse events that may be anticipated for a subset of the study population. We've already heard about the concept of a safety surveillance plan, which is described in the FDA guidance, which lists these anticipated events that won't be reported as individual cases to the IND, and also may include what we heard already about expected serious adverse reactions that are listed in the investigator brochure. So adverse reactions are those adverse events that have some basis to conclude that there's a causal relationship with the occurrence of that adverse event in administration of the product. If those adverse reactions are reported as serious events in clinical trials, they may be listed in the investigator brochure and considered expected for the purposes of expedited reporting to global health authorities. In order to meet the guidances globally for the SARS that are reported in the IB, at this point, I think those lists for products in development are limited. But for those SARS that are listed in the investigator brochure, those should be also noted in the safety surveillance plan and be monitored for cases that are different in nature than what's listed currently in the IB or are occurring at a different rate than is listed in the IB. So with our AbbVie projects, we've searched for the types and frequencies of SAEs in similar patient populations that are being studied in our clinical trials, mainly in comparable clinical trials that have a patient population with a background medication regimen that's as similar as possible to what's being administered in our trials. Also registry studies, and to some extent, claims databases. So potential sources for the clinical trial data for us have been advisory committee briefing books, health authority product approval summaries, also our own internal sponsored data sources, and publications including registry reports. But other potential sources include, we heard mentioned, the Transcelerate Placebo Standard of Care database, potentially electronic healthcare records and claims databases, which I mentioned. Also, you may consider using regulatory reporting systems to identify the most frequent events being reported in particular patient populations, and then, if relevant, the prescribing information of background treatment medications being administered in the clinical trial. So the participating product safety teams at AbbVie developed a safety surveillance plan for their products, which included the expected serious adverse reactions that were listed in the investigator brochure, as well as any disease-related events that closely were associated with study endpoints that were in the protocol. And then these two categories of events would not be reported as single case reports globally. But the other anticipated SAEs would be specifically addressing the FDA guidance. So we searched for public information, as I mentioned, and also our relevant internal databases to identify the most frequent serious adverse events in similar patient populations as would be in the clinical trials that we would be conducting. And we took all this data and merged it. And one of the approaches we used was to develop a heat map where we looked at the detection of the most frequent SAEs that have been seen across multiple data sources for those patient populations. So you can see in this graphic that those events that appear in red or orange would be the ones that were most frequently reported across different data sources. So for the AbbVie pilot projects, we have tried to do this blinded review using a couple of different statistical methodologies to look at how the rates in the ongoing clinical trials compare to historical reference rates, and when they meet a threshold based both on statistical judgment and medical judgment, that we feel the events merit being referred to a safety assessment committee, that those events then are referred over to an internal safety assessment committee, which operates under a charter, and that charter has been developed to mimic, in many respects, a data monitoring committee charter. And that internal group then may review that data in an unblinded fashion and make a decision about whether a threshold has been met for IND safety reporting. If an IND safety report is recommended, then the data monitoring committee would be notified and the product safety team. So some of the challenges we've had is, first of all, I think identifying the search strategy for the events, for the safety surveillance plan. You know, whether to use a preferred term search strategy or grouping of preferred terms or high-level terms, getting at more of a medical concept level, or possibly a narrow standardized MEDRA query, for example, are different approaches we've considered. Then, based on those search criteria, we try to come up with an estimated background reference rate, looking at multiple data sources and looking to see how much variation there is across the different data sources that we've looked at. Obviously, if it's relatively narrow and the rates are fairly consistent, that is very helpful. But then we select a reference rate based on our examination of the different sources. And then we assess which serious adverse events are likely to be reported in greater than 3 subjects in the clinical trials, depending on the patient numbers and exposures that are planned. Because I think, as was mentioned in the previous talk, I think small trials where you have few SAEs probably don't meet this criteria often. So we're looking generally at larger trials that have more serious adverse events that might meet this criteria. So then we determine among those anticipated SAEs that would be occurring at higher rates, which should be in the safety surveillance plan. And some of the questions that we ask during the review of the data are the events only anticipated for a subset of subjects? For known consequences of the disease, how specific is this event's association with the disease? Is there an overlap of the anticipated event with a potential risk of the study product? And what is the capability to assess if the event is occurring at a higher rate than anticipated? So some of the additional challenges we've had are complexities when a product is being developed for multiple indications. So in this particular case, you need to have multiple lists of anticipated events because sometimes the background reference rate can be quite different for different study populations. And this does make the execution of the safety surveillance plan activities more complicated. Also, we've had questions about the granularity by which events should be reported as IND safety reports. Typically, traditionally individual SAEs have been reported at the level of a MEDRA preferred term. But what we've been doing in our safety surveillance plan is really, reviewing the events at more of a medical concept level, which means that they're being identified by preferred term groupings or high-level term MEDRA groupings or possibly narrow standardized MEDRA queries. But this does lead to issues with trying to develop the reference rate because not always is the full list of SAEs available in public documents for different sources. And so, we can't always be sure that we're, comparing apples to apples, as it were. And also, even if the rate is reported as a group term in public documents, all of the preferred terms that make up that preferred term grouping are not necessarily provided. So, and as was also mentioned in a prior talk, the rates of anticipated SAEs could be influenced by factors that differ among programs like exclusion-inclusion criteria and investigative site countries, which again may not always be evident from public documents. So another approach is to use electronic healthcare records or claims databases to estimate reference rates. There are challenges with this approach as well because these databases use ICD coding as opposed to MEDRA coding that is used in clinical trials. There's also questions about the matching of the patient population with those in these databases. For example, for oncology trials, there's difficulty identifying similar patient populations in regards to tumor histology or greater molecular profile, especially in claims databases. Also, there may be differences in the approach to recording diagnoses leading to hospitalization in these particular data sources compared to clinical trials. And events that meet serious criteria in clinical trials, generally you're looking at events leading to hospitalization in these other databases, and that may not necessarily match up with events that are being reported in clinical trials as serious. So all this can be pretty labor intensive. But there are, I think, some opportunities for the future. In terms of identifying anticipated events, I think we could consider aligning, relevant stakeholders and elements applicable across multiple product development programs, in particular, known consequences of the underlying disease, for example, as well as events that are common in the study population. Also, events that are known to occur with drugs that are included in some of the standard backbone treatment regimens for a lot of clinical trials. So these event databases could be updated annually with relevant information with alignment across multiple stakeholders. Also, I think there's opportunities for better estimates for expected event rates with collaboration to identify potential data sources early in clinical product development. Some of the ideas are advancing the use of electronic healthcare records, which provide more granular information than claims database sources. Also, potentially developing a standard of care synthetic control arms, which use similar inclusion-exclusion criteria as what are being employed in the clinical trials. And then consideration for linking via a third party clinical trial patient data and mainly electronic healthcare records to derive comparator groups. So I just want to thank everybody at AbbVie who participated in these pilot projects and all their work on this. And I would turn this over back to the moderator. That's the conclusion of my remarks.
Thank you so much.
Yeah.
Please join us. So that was a great presentation, sort of highlighting a lot of the challenges, the sources, the issues of coding, the seriousness of the events, all of which are ones that we would like to discuss. So I think I'm going to start. We have a couple of questions, and I'd like to start with Adrian, and then I'm going to go a little out of order to maybe talk about your experience in this. We know there are many anticipated events in a population, particularly in elderly populations. And what has your experience been in terms of data sources and the ability to really identify a sufficient number of events with sufficient specificity to undertake the way we anticipate reporting these?
Well, I think for us in particular at Alnylam, we are dealing with many of our products with rare diseases. And I think the rare disease population really presents unique issues to figure out what are the anticipated events. Those are often diseases that are not well studied until there's a chance to study a treatment. And so one of the things that was brought up in the previous talk is the idea that the comparator groups or what has been done in previous trials to sort of sort out the background rates is very helpful, but often not fully disclosed, so that we can find those background rates. Because if the issue is felt to be in the background, it's sort of not part of the drug adverse event profile, and therefore not disclosed. So I think that's a rich source of information that we perhaps have not mined quite as well. And then, of course, another, you mentioned elderly, but I do think that as a pediatrician, I have to mention that, we also look at diseases across an age spectrum, and we have to be very cognizant of where to find that kind of background information and what, we have these sort of artificial age breakdowns, but that may not really be appropriate to what we're looking at, and it may not be accurately reflecting what those background rates are. So I think there are really unique challenges. We've also attempted to use electronic medical records and claims databases, and we found that in the rare disease setting that often the diagnosis or the coding is very inaccurate. So another sort of watch out when you use these databases, that you have to evaluate the source of the information before you can actually use it. So one of the things we've done is try to anticipate this and to set up things in advance. We've done for some of the rare diseases, we've done natural history studies before we've sort of gotten into our actual clinical trials in order to try to see what happens to these patients. What is their natural history? What are we planning to affect with our drugs? And so setting up disease registries or a comparison population in advance, if you can do it, can be extraordinarily helpful.
Thank you. Chris, you do a lot of, I'm going to turn to you, you do a lot of cardiovascular trials, so some of your events are actually your endpoints. But can you speak of the challenges in some of your populations that these, we often speak of the MI as the stroke as the obvious ones, but in determining those other anticipated events in those elderly populations beyond the cardiovascular?
Yeah, maybe let me start by also congratulating Barbara on a really nice presentation, I think, that highlights the complexities and the need for a plan that's specific to many of these variables, which I think are so important. And we can't really, in my sense is we can't really have a highly kind of structured requirements because of the complexity of these things. One comment that I might make, though, is I think we're really getting into trouble when we use either things like claims data or even electronic health data as a reference for safety for what we're expecting in clinical trials, especially when we're looking for anticipated effects where there may be only a modest difference in those events according to treatment groups. And I think we're fooling ourselves if we think anything other than a randomized comparison group will really provide anything reliable to understand those safety events. With respect to the outcomes, another context comment that I'd like to make is I think the most important thing we can do to understand what the effect of drugs have on kind of the anticipated common, really important effects is to have large trials with carefully defined outcomes that we reliably determine. And we had this meeting with Bob Temple and David Demetz and others a couple of weeks ago looking over the experience of the diabetes cardiovascular outcome trial experience, which I think is a really nice example. We're not going to be able to understand the effects on these critically important common events unless we have large enough, long enough trials to assess it. And I think the diabetes experience is one that shows how valuable that can be to really understand not only are we have safe drugs, but do we have drugs where sometimes unexpectedly, actually, we have beneficial effects on some of these cardiovascular outcomes. And then just three other brief comments that I think speak to some of the experiences that we've had around these issues. One is this issue of structured versus unstructured data collection, active versus passive. And I think this is critically important. If we really want to know the effects, for example, on myocardial infarction, we have to collect not in an unstructured passive AE approach, but we have to ask, did the event occur or not, systematically according to a definition? And we need to publish more of this information. But for example, we've had experience where when we have SAE reporting and redundant SAE reporting and structured reporting, that we, for example, had half the number of myocardial infarctions identified through SAE reporting that we have when we ask, did or did not these events occur? So I think we're fooling ourselves if we think through passive AE reporting that we're collecting accurate information on some of these common events. Another example I have is a DSMB that I chaired where we had a drug that was anticipated to potentially have liver safety issues. And when the liver safety was collected in a highly structured way, systematically collecting information like liver function tests and looking at it with a drug-induced liver injury expert panel, we saw a clear and compelling evidence of liver injury, which when we looked at the SAE reporting, which we never would have detected. So again, it's an example where the data is going to be only as good as the kind of structured approach to collecting it, and I think that's really important. And it alarms me and I've seen this, and I hope it's not happening anymore, when data is collected for outcome events that are important through a passive approach of AE reporting, identifying those potential events, and then collecting them and thinking that's a reliable way to collect events.
But can I push a little bit more on this? So in a trial, how would you, recognizing we don't want all the events individually, how would you go about it as you're starting a trial to think about what to do about these anticipated events in the population and how to know whether to report them?
So I think the first thing is to collect them in a structured way. And of course, this depends on the drug and the population and the size of the trial, but any trial in a population that has common cardiovascular outcomes, because that is the most common cause of death and disability that people experience, that older populations experience, those should be collected, I think, in a structured way. Now, if they're not, then and sometimes they won't be if in an intermediate population, let's say. Then I think we still want to have some type of adjudication of the events about the reliability of how they fulfill kind of standardized definitions for these events as one of the important approaches.
Okay. Mary, do you want to comment sort of from
Sure,
our perspective?
Well, I am the safety deputy in our division of cardiovascular and renal products. So I'm going to describe a little bit about what it's like from an FDA medical reviewer when they get these IND reports. I took away some great takeaways from Dr. Wittes' presentation because it really kind of highlights where FDA might fill in the holes with these because we have sort of access to lots of information that the individual sponsors do not. But just for a reality check, the way our current system is set up, it's not even real easy for that sort of information to be gleaned across our IT systems right now. We are starting some initiatives where we are going to be able to look at things in aggregate across therapeutic areas, across indications, and that I think might help fill in the hole of that sort of absence area that has been identified that individual sponsors don't have access to. Medical officers, to deluge them with reports that don't mean anything increases the likelihood that they don't look at anything. So, people will sometimes just almost throw up their hands and not be able to look at case by case by case and try to make some sort of safety conclusion based on what's being sent in. Medical officers have different review practices. Some have lists of safety issues that have traveled over since, preclinical, since the IND opening, and they will compare reports to that list. They'll update that report. Some are less diligent about keeping a running list. So you can see that sort of environment makes it difficult to look at case-by-case things where they can't interpret whether it's an anticipated event or not. I think one of our guiding principles today probably is, and I think most of the reviewers at FDA probably agree, that the sponsor is in the best place to integrate safety knowledge. You know, they have the knowledge from preclinical to early phase to ongoing studies. They are knee deep in the program, whereas an FDA reviewer will have lots of different programs. And so I think we very much rely on the sponsors to do that curation of events to send to us so that we can sort of make sense of them from our point of view. I think I've heard the theme of sharing data on background rates for certain things that people often come across in the post-marketing setting when you're wanting to try to compare what's happening in the treatment group to a control group. I think we have a lot of interest in that as well. It helps in the IND pre-market setting. It would help us in the post-marketing setting. And I think we've thought about doing this a little bit with Sentinel because that's our sort of EHR claims tool that we have access to. But I hear the pain about that. I mean, you're only capturing certain events where patients are hospitalized. It certainly does not apply to the gamut of things that you come across in a clinical trial. But I still think that it's a very rich source of data that we should explore and figure out what can be used and shared. I think that there's an opportunity here to bring lots of people together who have these data. I mean, everybody's doing their work in their corner. They probably all have little, nice pockets of information. If we could establish a library and refine those event rates as practice treatments changes, as medicine advances, I think that would be a really, really good initiative to get off the ground for everyone. And along those same lines, I think it would be important to, and I don't know if this is the case or not, whether some work has been done to compare how that worked for you. You identified this historical rate or anticipated rate of MI in this population. How did that ultimately end up comparing to what you saw in the trial? And do that for multiple data sources. I think that would be very informative and help maybe refine how you approach identifying anticipated events.
Barbara, you mentioned that in your pilot you sort of looked across a number of different sources, and as you were doing that, so can you sort of speak to, was there such variability that it became very difficult to even get to a rate that you might think was reasonable? And also sort of in terms of the anticipated events, the cardiovascular or outside the cardiovascular area, is there places where it was easier and more reliable, or in the oncology area because we have SEER reporting and everything? Is it more? Can you speak to that at all?
Yeah, I work mainly in immunology. What can I say about that is that we did try to go first to more clinical trial data sources, and the placebo rates often can be highly variable because the placebo arm is often has fewer patients than the treatment arms. And so the rates can be quite variable because the number of events may be not so large, so that can be a bit of a challenge. So, in looking at the events also at the active treatment arms, especially if there's been no reason to believe that active treatment is associated with that event, we look at that as well to try to get an idea of the range, in the patient population. Also, registry studies has been where we've gone a lot to look at, so we have reports from various registries we have access to, and so I think that's been a great source of information for us is the registry data as well. So,
can I just comment though again about this? I'm a little bit surprised because there's lots of data that, for example, simply the act of getting consent from a patient to require it as a requirement to be in a clinical trial creates a highly selected population that I think of on average. Once you get consent, that patient has half the rate of some of these major adverse outcomes than an unselected population. I think we're totally delusional if we think that we can use an unselected population as a reference rate for patients in a clinical trial. And I think we have lots of data to support that. So it may be relevant to know what that background rate is, but we should be very careful in suggesting that that's an appropriate rate to compare adverse events in a clinical trial.
Well, I think what we try to do is look at a variety of data sources. Clinical trial data is our first preference, if we can, because I agree with you, it's the most aligned with what we're currently doing, looking at the rates in another clinical trial. So, but I think what we look is across various data sources and see how close are the rates. Actually, you know, a lot of times the registry rates and the clinical trial rates aren't so far off from each other in our experience for some of the things we've looked at. They've been pretty close sometimes. So, you know, if you see consistency across multiple data sources, then that gives you more confidence, you know, that you're probably on the right track. But if you're seeing big discrepancies, I agree with you, then you have to think about, like, why is that, you know? And some of the problem is with the clinical trial data sometimes is that the number of events are so small that there's a lot of variability. You know, you need to get a much bigger database of studies pooled together, I guess, to really look at a rate that is more accurate. So that's some of the challenge there for us. Yeah.
It is a challenge, and I would agree there, but two comments. One is that sometimes that clinical trial population is exactly very close to the clinical trial population you're dealing with. So it is a good reference rate for your particular trial to see if your rate is different. The other thing is, you know, we're talking about sort of signaling really within the earlier points of a clinical trial. And I think at that point, even if your reference rate is a little low, it's okay, because you'd rather be on the conservative side and pick something up earlier than have a more general population rate that where you may miss that signal within your clinical trial. So an underestimate in the background rate may be acceptable in this particular case.
One other thing, I mean, we'll get complete NDA packages where the trial's done and you see these imbalances and you're still, you know, I'm not sure, like, why would this drug cause this? So there's a lot of uncertainty once those data are all put together. So to make these kinds of decisions before that even happens, I think you have to understand that there's going to be a lot of fudging and judgment and eyeballing it. So until we get more precise background rates from whatever source we determine is the best one.
I guess the other thing, just before maybe we open up to questions, we had talked about, I think Bob Temple said there's too easy and too hard, and the other hard is sort of the events that might have been identified or anticipated with your drug, but whether the severity or frequency is elevated. Is that also a challenge, or is in sort of what we've been asking sponsors to do in this guidance, or is it really the anticipated that is our true challenge?
I mean, I think that's a critical point. And I go back, Salim, we were talking about in developing fibrinolytic therapy, there was a clear, I mean, we knew the main trade-off for the reduction in mortality related to the myocardial infarction itself was intracranial hemorrhage. And it was an anticipated event, but it was the critical event to be carefully monitoring to assure that the drugs had both safety and an appropriate trade-off. And what we tended to do was to have the DSMB very carefully tracking that, and even with predefined guidance on what might be appropriate trade-offs. But I think sometimes the anticipated events sometimes they're the most important in terms of assuring ongoing accurate assessment of safety and the trade-off of safety and efficacy. I mean, maybe that's an example where the DSMB is the key mechanism, and we don't need to involve the guidance that we're talking about.
Mm-hmm. Anyone else? Are there any? We have a couple of minutes. I don't think we're out of time. I would be happy to take some questions.
Maybe that microphone.
Yeah, I think there's a mic. I think they're just about to turn it on for you.
I mean, the discussions here show how difficult it is to estimate background rates. But aren't we creating a huge monstrosity for nothing? And what I mean by that is we have a superb methodology called randomized trials. And we're now going to the worst methodology called historical comparisons. Aren't we being foolish with this step? Shouldn't we just step back and say, really, no matter how hard we try, we can't predict background rates. Let's even take conditions which are the primary, in which certain outcomes are the primary endpoint, like mortality in a cardiovascular trial. Our prediction of event rates, for which there's a ton of data, is not that great. So how are we going to then take adverse events, which haven't been systematically documented in the literature, and come up with realistic rates? And Chris made a very good point. The people who you select in a trial isn't your average registry or your average clinical thing. I personally think this whole thing of unanticipated excess rates should be thrown out of the window completely. In fact, down the toilet, it should be flushed out. Instead, let us focus on the strength of the randomized trial, which is the randomized concurrent comparison. And in every drug will have side effects. I remember my mentor, Richard Peter, telling me, if a drug doesn't have side effects, it won't have effects. So if that is the case, you have to balance side effects with effects every time. I mean, you can't just put side effects alone. And goes back to the thrombolytic trials. There was a clear excess in intracranial bleeds. They emerged early, but the reduction in mortality took longer to emerge when the GC trials. If you only did this and looked at only safety, you shut down those trials early and we'd miss the benefit. So I have two thoughts for discussion. Let's get rid of this thing of unanticipated higher rates of events. That's just a fool's game. And it's just a, create work situation. If you want to politically create work, let's do that for unemployed safety reporters. But the second part of it is, I think looking at non-randomized data loses the strength of what we're doing. And we should look at it in the context always of potential benefit. I'll leave it there.
So does anyone want to make a comment? Or?
Do you mean throw them out, i.e. we don't need to even consider reporting them in an expedited fashion? I'm just wondering how to.
Absolutely not. 99% of them are irrelevant.
So you just keep them in your database and
let it be collected.
Right, but don't make any decisions along the way.
Mm-hmm. And they view it with all the criteria, seriousness, all that kind of stuff, but also compare it across the two groups.
Which is your best
that is
comparison?
Your best comparison. And they weigh it against your benefit. There are examples that Chris and I have been involved in. When we did the studies of fondaparinux with OASIS 5, there was higher rates of reports of catheter thrombosis. And the regulators, not the FDA to their credit, but some European regulators, demanded we unblind data. The DSMB chair, Peter Slide, was tough. He said no. And he managed to persuade them. What that wasn't telling us was we had a 50% reduction in serious bleeds, including intracranial bleeds, and nothing that we had anticipated was a 25% reduction in mortality at that time. So if we had just reported catheter thrombosis, although the excess was 10-1 on a rare event, and it was real, we would have shut the trials down, missing a substantial reduction in bleeding. Okay. Oh, okay. All right. So now, you want me to start all over again?
As you're walking back, I just want to make one point, though. I mean, one of the reasons that we're talking about this is this was a method potentially not to unblind everything to make your comparison. So there was sort of two approaches. One, you unblind and make that comparison between these events, or you identify a rate up front and only unblind when you exceed that rate. So this was a way of trying to balance our need, or our desire really, to have more reporting that was more informative without jeopardizing what we recognize the integrity of the trial. So we need to put that in context. It's not that we don't think randomization, or we're saying randomization is great. We just don't know what to do with trying to unblind to get at this issue.
I think the solution you ended up was the wrong solution, unfortunately. Okay. You know, I understand the intent. Mm-hmm. And what I think you should do is the unblinding should be done not by the sponsor, not by the investigator, but it should be some form of regular safety reporting, and ideally by the data monitoring committee, mm-hmm. because they're seeing the totality of the evidence. It could be that one adverse event is going up, but a whole lot of things are going down, and they're putting it in the context of the efficacy. Now, yeah, I think you're losing the strength of randomization. You're losing the totality of the evidence, which is the context with the current method. And it's a huge work creation effort that has now led to a huge machinery. And the machineries are not just whether FDA is drowning in paper. The IRBs at each center are drowning in paper. And our IRB and all the IRBs in Ontario, in Canada, have said, we don't want to review them, but if you want us to review it, it's $300 per case that you have to review. It is a way of pushing back and saying, this is nonsense and silly. We're not going to do silly stuff. And the other part of it is, in a study that we are coordinating around the world called COMPASS, we have to send monitors to make sure that the investigator sent it to the IRB and there is documentation. And there is concern that if we don't document, when the auditors from FDA come, we will be cited for lack of oversight. Now, this whole rigmarole is useless. And instead, let's just do decent big trials and get a good, sensible group of people to review it. That's the easiest way to do it. But yet, yeah.
I think there is a part of that in what is being described here, and that is the use of the Safety Assessment Committee that can unblind data, that doesn't make independent decisions about shutting trials down, but they work with, I think your slide showed this, they work with the DMV, and so that they're not just saying, oh my gosh, we see catheter thrombosis, shut it down. You can't, that's not the right decision to make. The right decision to make is to go to the people that can look at both sides, look at benefit and risk, and say, do we need to reconsent? Do we need to, I mean, that's not a great thing to have to do, but do you need to change monitoring, collect more information on the events? But I think I understand your discomfort with using historical rates because they are really all over the place. But I mean, and I agree with using the randomized data if you can. And I think that model is built into some of the things that have been proposed here.
Salim, I think the whole point of the changes was to do exactly what you want. What we were getting was, just as an example, every heart attack would come in. And it would go to all the IRBs, it'd go to all the investigators. And, you know, in a population over 65, that's not very helpful until you break it down and look and see if the rate is higher. So what it calls for is not reporting those until and unless there's some reason to think the rate is higher. And as was discussed, that could be because the overall rate's higher than you expected. But that doesn't mean you should then report it. That means you should then unblind it by a data monitoring committee or something, or a safety assessment committee, and take a look. The trouble with, I'm not sure I understood what you wanted to do, but this is something you do during the study. It's not at the end of the study. At the end of the study, you're going to look at all that stuff. It's trying to anticipate that the drug is doing something bad. That's sort of a responsibility you have to the patients. So you need to get a smell early when it looks unbalanced. And how to do that, that's one of the main things we're talking about. How do you know when you've passed a threshold or when you should look? I guess one possibility is to take almost any serious adverse effect and have a data monitoring committee look at the rate of all those unblinded. And that's one perfectly plausible solution. And it's not incompatible with what we suggest, but that's a lot of work. And we also, by the way, just want to mention it, it's not necessarily only one study we want people to look at. We want them to look at the whole database. So if there's 5 studies, you know, in depression going on, we want them to look at all of it, and how to do that is the problem. But not sending them in before you do that is what we were trying to, that's what we were trying to accomplish, the very thing you're worried about, so.
Thank you.
Thank you. Robert Baker from Lilly. I have, I hope, a shorter question, which is, I'd be interested in the panel's view at a bit higher level. Are we aligned on exactly what we're trying to accomplish? Because I think that can help us to narrow or focus the tactics. And specifically, I asserted when I spoke that this is about subject protection within our trials, within our programs, whilst maintaining the integrity of the blind. And I think if that is indeed what we agree on, we can have tactics that might be more manageable. But I could also envisage that the agency might be interested in maybe lower severity things and more contemporaneous as you're looking at other programs. So I think it'd be useful to know whether really, if we get this right for the kind of serious things that would really shift the benefit risk for our subjects, that we've done enough.
I'll defer to Mary, but certainly this is all about serious adverse events, and and sort of looking and getting information that is relevant to the safety of the patients earlier if we can. And when it comes in as sort of a single event and that analysis is not done and it's not done across the development, it's hard for us to get that signal early. I don't know, Mary, if you want to comment further.
Of course, the overarching principle here is to protect patient safety, but in order to do that efficiently, you have to put some thought into what you think is important. I mean, I don't think anybody is interested in shutting every trial down for every SAE. I mean, and a lot of times you don't need to do anything. You just, it's something that you might have you might have thought of from the preclinical model, but just sending everything in without filtering it down and trying to come up with a list of these things is not a good way to protect patient safety, because it's just paperwork.
Yeah, just to be clear on maybe more clear in the question, sorry. It's that we will, of course, when the trial ends, we'll have data, we'll have the randomization, you'll see that. I can imagine tactics that we could use in unblinding groups or even the DMCs that'll keep pursuing this at a subject level, but I guess how high is the priority that you're seeing that extremely quickly versus that we're protecting the subjects? Because if it's the former, then I think that we're going to have to cast a wider net.
I'm sorry, it's a former that we're seeing it very quickly or that we're protecting subjects? I guess it's hard for us to divorce those two, because I think that's what, safety reporting is about. It's just that it's both of us working together towards that same goal and trying to get there. So I don't, we're not asking for these early just to see them early. We're asking them early because we want to make sure that we're doing the right thing for patients and for the trial. And I think in many ways the thought was we're not asking for them early with this guidance, and maybe that's not clear, because we're not asking you to report them as they're happening if they're anticipated. We're asking you to look at them and keep monitoring them and report them when now we are seeing a difference that we need to pay attention to, because it could be a patient safety issue. So I think that's the goals we were trying to get at. Maybe we have not done that, and that's sort of why we're in this room today, to figure out how to get it, optimized.
And I'll just reiterate, I think it is really important that there's this somewhat of a shift to the expedited reporting issue to, in these anticipated events, the topic of this panel, that there's more of a focus on that, on the common serious consequences that our patients are experiencing, or events that our patients are experiencing, and making sure that signals aren't arising, that there's a substantial difference in those that we should be identifying for patient safety.
Yeah, I think one of the questions I've had is exactly what was brought up, is for expedited reports that, when you're looking at these events that may not rise to the level of stopping the trial or even, a protocol amendment with different exclusion criteria or different safety monitoring, where is that threshold for just informing investigators there is a possible causal association with this product that they need to be aware of and take into account when they're dealing with this individual patient before them and with their, constellation of, medical circumstances, versus rising to the level that you think you've gotten to the point that you need to modify the trial, which we know, at that point, you really do need to look at the benefit versus risk sometimes, that the Data Monitoring Committee is in a better position to do.
See what you're saying, the sort of threshold issue in terms of how
Yeah.
fast and when you bring it in and when we're recognizing. I know we're a little over time. I'm going to let 2 short questions maybe.
I'll keep it short.
Okay.
Right, Bill? I think we're a lot more aligned than the discussion sounds here, but we have to remind ourselves we're talking about anticipated events primarily. So events that where you need an aggregate analysis, you have some information here. At Merck, we try to remind ourselves periodically that we're looking for the wolves, not the chipmunks. So as the reviewer pointed out, even at the end of the trial or the end of the program, where you have all of the data clean, locked, integrated together, it's still hard to find the chipmunks. So we're looking for the wolves here. And for the discussion that was brought up before about, registries maybe not being useful or some other data source maybe not being relevant, it depends. Sometimes it may be useful. I think you should gather all of the available information, taking into consideration the relevance to the ongoing trial. So, for cardiovascular endpoints that have to be adjudicated, maybe it's not useful to look at a registry. But there's a lot of endpoints where that'll give us a more robust understanding. So, and we shouldn't overpromise to anybody here what we're trying to do, right? We're trying to find these wolves again in ongoing trials. So, if we don't have relevant information about the background rate, then we can't take advantage of some of these more quantitative methods. But if we don't have a lot of understanding, then it should be a little bit more difficult to cross some threshold anyway. So it's not really a question, but I just wanted to emphasize that we have to keep reminding ourselves what we're really trying to do here. We're not trying to find a difference in a very rare event. That's the type A and B events, the single or small clusters.
And final comment.
Hi, Asif Haq from AstraZeneca. So my question or a thought for an alternative approach, whenever the sponsors come to the FDA for guidance on a suggested plan for investigation study, there are also discussions on what events should be reported or what events should not be reported because they might be endpoints. Now, I think we can define that further because the common question everybody's talking about is incidence rates. So perhaps there could be a discussion or some guidance from the FDA at that point that these are particular events and these are the event rates that we have seen. And perhaps if the FDA can, let me just add one more thing for them to do is to figure out or align on a better incidence rate that based on their global knowledge and then provide that to the company saying, this is your threshold. This is what we want you to do. Because currently we're all searching for the same question. Where's the threshold? Who defines it? And which data source do we look at? Because every data source you look at is going to be different. And everyone looks at it from a different angle. And again, when you look at a patient population, you're talking about severity, grading, cardiac failure, et cetera, et cetera. So everyone will have a different set of brackets to do. So is that something that can be thought about when a sponsor comes to you at the design phase, that you with your background knowledge on and all the background information that you have, set that standard at that stage. Because then that will really help us also to work from a standard, rather than build our own standard, which might be different for everyone.
I can tell you that we do not have the magic background incidence rates. I mean, but we do have access to different things that other people have access to. And so I think we would be certainly willing to work with you when you came in and said, these 5 events we think are going to happen in our, patient population based on X, Y, Z. Using these data sources, we're going to, our range of expected incidences, blah, blah, blah. I think we would welcome working with you about, sort of looking at how you came up with those rates and agreeing or disagreeing or
and also publish them so everyone has access
to that's
what
I mean. A library of background rates would be lovely. Yeah, I mean, I think these are
because we all struggle with that whenever we have to look at an aggregate report. Which database do we look at? Do we look at USRDS? Do we look at this? Do we look at that? So, thank you.
Thank you. We're a couple minutes late, but we're on break now until What, sorry, one second?
What? 1 second. I think this discussion's going the wrong way. We're going to create a library, we're going to give you a library, and we know it's terribly unreliable. Get rid of this idea and stick to the randomized data. The request was not sensible. I'm not sure the response is going to be useful.
Okay, so we have both views on the table here. Well, thank you, everyone, panelists and presenters, for a great discussion.
Great, thanks to Jacqueline and all of the panelists. We're going to go ahead and take a 15-minute break, or actually 10-minute break. We'll start back with the next session at 11 o'clock. Thanks. Okay, we're going to go ahead and get started. Can I ask you all to please make your way to your seats? Welcome back to our second session focused on maintaining trial integrity with unblinding and clarifying the flexibility in choosing the entity responsible for aggregate analysis and IND safety reporting. We've heard quite a bit of discussion already so far about the responsibilities of these various groups and how to do this. I'd like to now introduce our moderator for the session. I know that. Okay. I'll go ahead and introduce our moderator for the session as Professor and Associate Chair and Director of the Collaborative Studies Coordinating Center at the Department of Biostatistics at University of North Carolina, Lissa Lavange. Lissa, can you join me up here? And then I can ask the panelists to go ahead and make your way up on the stage as well. Thank you.
Thank you, Greg. I'd like to introduce our two speakers and panelists. We have, joining us on stage, AJ Singh. AJ is the team leader in Safety Evaluation and Risk Management Group at GlaxoSmithKline. Our second presenter, Tom Fleming. Tom is the professor and former chair of the Department of Biostatistics at the University of Washington in Seattle. And we have 5 reactants, 4 in person and 1 hopefully joining on the phone. We have Amit Bhattacharya, Vice President of Biometrics at ACI Clinical. We have on the phone, if she can join us, Erin Bahula, Associate Physician in Cardiovascular Medicine at Brigham and Women's Hospital, an instructor of medicine at Harvard Medical School, and an investigator in the TIMI study group. We have Alyssa Malkin, Medical Director for Pharmacovigilance at BioMarin Pharmaceuticals. And we have Dave Demetz, Emeritus Max Halperin Professor of Biostatistics and former Chair of Biostatistics and Medical Informatics at the University of Wisconsin at Madison. We have finally Matt Sukup from the FDA. Matt is Deputy Division Director for the Biometrics Division that oversees pre-market and post-market safety evaluations in the Office of Biostatistics, Center for Drug Evaluation and Research. So we have a really wonderful, distinguished panel. We have a plethora of statisticians, and we're going to get into more of the nitty-gritty of how we can figure out this more efficient and effective IND safety reporting. This session has primarily 2 objectives. We want to look first at the challenges related to unblinding data to determine if there's an imbalance in these events between treatment groups, as we discussed in the first session, and also whether that could be maintained by a group other than the DMC, whether the trial integrity could be maintained in spite of this unblinding. And then we want to talk a little bit about the Safety Assessment Committee, how it differs from a data monitoring committee, how those 2 might work together if they are both maintained or in a particular trial. So AJ is going to start off with a presentation from the industry's perspective for us.
Hi, Lissa. This is Erin Mahou. I just wanted to confirm that I'm on.
Great. Thank you.
Thank you, Dr. Lavange. Appreciate it. So I'm AJ Singh from GSK, and Dr. Virtus, I'm going to have to approach your AE to our vaccines office. So hopefully it's not serious, but we'll touch base later on. But again, also my appreciation to Duke Margolis and FDA for really allowing, I think, an open discussion about the extent and proposed safety oversight guidelines in the context of the INDSR reporting structure. I think it's fair to say that all of us, whether we're in academics, the regulatory bodies, ethics committee, or industry sponsors, are facing mounting challenges with this safety oversight. And I was sort of thinking about this, and maybe we should celebrate these challenges, because now with the advent of targeted therapy and immunotherapy for cancer, and for individualized therapy for rare diseases, we can really study patients we couldn't study 10 years ago, 15 years ago, and make a difference in their lives. But these patients, as you all know, come with significant comorbidities. Now are on litany of medicines which we would exclude before, and many will have underlying endocrine compromise, which makes the evaluation and assessment of causality of treatment-emergent adverse events much more complex. Especially if you look at some of the ones listed at this is a very small group, obviously. Ones like liver enzyme abnormalities or renal function problems, which have a multifactorial etiology. Now, the last one is close to my heart. I am a nephrologist, and is one of the hypotheticals that was presented in the guidance document. So if I could just ask for your forbearance here and just present somewhat extended to this hypothetical that was in the guidance document. A not unusual scenario we may face, all of us, I think, would be a cluster of 5 events of acute renal injury or acute renal failure among 200 patients in a cancer trial. Now, hypothetically, what I've said is that in a non-clinical evaluation at high doses, this drug was associated with evidence of tubular injury. Each of those 5 cases came in unrelated because there was another risk factor, dehydration, diarrhea, or previous exposure to nephrotoxic chemotherapy, not uncommon in this population. Therefore, when we do the pharmacovigilance assessment, what we would likely say is that the cases had other risk factors, and in the vernacular of the pharmacovigilance world, we'd probably say they were confounded, with apologies to my epidemiology friends here. But I think the caveat here is that it may be precisely these vulnerable patients who present first, who present at the lowest exposure, at the lowest dose, i.e., if there was an effect on GFR, i.e., an elderly dehydrated patient may be much more likely to manifest the clinical complication of tubular toxicity than a 25-year-old that's well hydrated. Secondly, even though each case was deemed unrelated, I think all of us would have done that. We as a sponsor and at the regulatory bodies have sight of the cluster of events. And thirdly, we had a very, very nice discussion that even though if I review the literature, renal compromise is very common in the cancer population. And historical controls serve, I think, as a good barometer, but really the best comparison would be the drug versus contemporaneous control. I think we all realize that. And I must confess, when we have these type of discussions, which we do all the time at sponsor level, and I'm sure at the regulatory level, I found myself muttering under my breath, God, I wish I knew the drug allocation. Just to be reminded by my more sagacious statistical colleagues and clinical colleagues that we need to maintain the trial integrity. And really that's the question for today. In terms of what are the challenges of maintaining trial integrity with now routine unblinding of safety data, or more specifically, the SAC concept, and to have them review SAC committee, have them review unblinded SAEs across the development portfolio within the context of the INDSR evaluation process. Now, this slide we've talked about before, not to belabor it, but we've made a distinction here, events which can be reviewed on isolated cases versus those that clearly require an aggregated review. And amongst the latter, I've just put into the categories that have been discussed: emerging new signals, that is previously unexpected events, or perhaps a more challenging concept of higher than what we expected rates of already identified events that were suspect or anticipated events. And because this latter cohort, Group C that we are calling it, requires comparison to either historic or contemporaneous controls, the proposal is for a routine unblinded review by this oversight committee, the so-called SAC. Which gets to me to really the first challenge. If we constitute SACs across the developmental portfolio to look at all serious adverse events, that will, for many companies, require a lot of data being unblinded and a lot of colleagues that will have at least sight of the unblinded data, whether it be in the aggregate level or at the patient level, from the data managers to the statisticians and the oversight committees. And this does reflect truly a paradigm shift in how we do in-stream safety analysis. Not to say it's anything wrong, but it does suggest for us a big change in how we would do our in-stream analysis. Now, one can imagine that these colleagues on the oversight committee will likely be looking at a lot of signals, sometimes with small numbers and sometimes with incomplete data. And hence, on occasion, and it's possible even more so than on occasion, they may require supplemental information. So let me just go back, if I could, to this hypothetical case of the 5 reports of acute renal failure, clearly an event that we're all worried about. And let's say there is an imbalance, a 5 versus 0, 4 versus 1, whatever we think is a bar that we think is at least meaningful. After data cutoff and after formal unblinding, the study team would have the benefit of contextualizing this SAE imbalance within the broader framework of trends in AEs that are relevant, as well as trends in labs, creatinine trends, whether they be summary data or whether they be shift tables. So depending on the clinical importance of the emerging signal, it may be possible that the SAC committee may request further unblinding, and I think it's possible that unblinding may extend beyond just SAEs to evaluate an emerging signal. Now, I don't think anybody in this room or any of us would argue that the well-being of the patient supersedes any other consideration. I think that's what we're all here for. But I think we would all agree, and the last conversation I think was very helpful, is that these considerations are often highly nuanced, right? And the decision to unblind beyond SAEs for a committee may be highly challenging because they're likely to be looking at multiple imbalances, small numbers. Many of them may be clinically important, and they may have incomplete data. So I think this is another one of the challenges that SSC will likely face as they look at interim data with incomplete information of potentially really important events where we don't have a lot of information and we're only looking at SAEs. I think Dr. Granger pointed out one case where it was the adverse events in which the liver signal came out. I think that could happen not infrequently. So sort of taking a step back, irrespective of the amount of data we unblind, if we have appropriate barriers in place, trial integrity should not be compromised. So whether we go with the IDMC model, where everybody is external, that's possible. And certainly there is precedent for companies having independent, parallel, unblinded review in certain situations by a separate group. However, the caveat here is that, at least at GSK, we have very small numbers that are doing that. And therefore, if you have small numbers of colleagues that are sequestered, it is fairly easy to maintain significant barriers. The practical logistics will get much more complex as the number of SACs and the attendant data unblinding grows. So I think, just to summarize, the effectiveness of these barriers will depend on the size of the company, will depend on the structure of the barriers, but also the constitution of the committees, with external committees being the most robust in terms of barriers. But I think there are some real consideration, and these were in the feedback to the docket, of why internal members may actually be beneficial. So this committee will be looking at a lot of signals, and we'll have to interpret it in the context of the population, but also the full non-clinical data, which may help to assess biological plausibility. And if data pooling is required, they will need a very, very in-depth understanding of the design and the results of all the previous studies. So in many instances, internal colleagues may be well-suited to sit on these committees. And secondly, we are recommending that the SAC provide a recommendation on INDSR submission, which is a major regulatory role with attendant effects on protocol and as well as ICFs. And sponsors may not feel it appropriate to completely relinquish these responsibilities. Skipping over this, I know I'm running close on time, but one other thing I would like to just point out, reading the responses to the docket, a recurrent and common request, I think, would be the best way to put it from sponsors, was a request to perhaps maintain some flexibility on the need for an SAC and the remit of an SAC based on the population studied, the stage of development, and a priori safety signals. And I think this is in recognition of the fact that most companies already have robust processes. So I'll take the last 30 seconds and just describe our process at GSK, and please, this is in no way to say this is an ideal way or ideal process. It is one paradigm. So the core of this safety construct really are the multidisciplinary safety review teams, which operate with guidance from and oversight by several other committees, all under the governance of our Chief Medical Officer, CSO, and Global Safety Boards. The safety panels are advisory. In the spirit of the SAC, we have 3 possible oversight committees which can do parallel, unblinded review if required. However, these are not mandated, with the exception of IDMCs, which are almost always required for pivotal or registration studies. The other 2, ISRCs and DRCs, are constituted for almost always for early phase products and with advice from the governance body and R&D leadership, and they can do parallel unblinded review. However, only a minority of our projects that do not have IDMCs operate under an oversight committee. Regardless, the responsibility of INDSR does stay with the core safety team, and they are encouraged to seek guidance from other panels. And one of the other panels that we have are formal safety panels, of which we have a lot of them. I've just listed a few of them. These are formal panels that generally have internal colleagues, but some external colleagues that are basically available to review emerging signals and help out the safety colleagues on an ongoing basis. So just one possible paradigm where there is a flexible approach. And with that, I think I'll turn it back to Dr. LaVange. I want to thank everybody, and really my genuine gratitude to the FDA and to Duke for facilitating this conversation, and to our esteemed panel for providing further context. So thank you.
Thank you, Vijay. And now, Tom, if you could provide us with your presentation. AJ, did I just call you AJ?
AJ's fine, yes.
Thank you, Lissa. So as AJ and Lissa have already pointed out, there's really a dual challenge here. And one is to get the timely and reliable IND safety reporting, but to do so in a way that protects the integrity of ongoing trials. I'm grateful to insights from many folks, discussions I've had with Lissa and Dave Demetz and Janet Wittes and many others on this issue. And in particular, I think Janet's article on the FDA final rule was very enlightening to me. As that article emphasizes, that as is in the guidance, there are really 3 simultaneous characteristics that we're looking for these key events: that they're serious, that they're unexpected, and then the art of this is that there's evidence to suggest a causal relationship, that they're not just adverse events, that they're suspected or certain adverse reactions. And so I found it very useful that the FDA guidance has these categories A, B, and C, and Janet's article talking about the kind of evidence that we would use for each of these to understand the drug's causal relationship. Categories A and B, if you have a progressive multifocal leukoencephalopathy, you only need a single event. Probably don't have to unblind it. If you have an MI in a young patient, you probably only need a few occurrences. To me, this isn't controversial in this setting in terms of process. Yes, we would have medical monitors. Yes, they would be monitoring continually. Yes, they would unblind as necessary to identify those cases. The challenging issue, as others have said here, is category C, these common events in an exposed population, MACE events in populations with moderate to high cardiovascular risk. There's no question, as others have said, that to understand causality here, you need to be able to compare the frequency in a drug group against a comparator group. And as both Chris and Salim have pointed out, that really needs to be a randomized trial, and it needs to be of sufficient size. This could be done by a data monitoring committee. It could be done by a safety assessment committee. FDA talks about these safety assessment committees as achieving timely identification reporting of SUSARs. So I'm going to talk about some important operating procedures of that SAC. It's going to be certainly important that it has insights from unblinded data from both completed, and here's the critical issue, ongoing trials. And it's important to recognize that in ongoing trials, we already have multiple oversight bodies that have overlapping ethical and scientific responsibilities. In a clinical trial, sponsors and investigators have key decision-making responsibility for design and conduct of the trial. Caregivers for the oversight of their patients, IRBs and regulatory authorities for approval of the ethics and the science and the ongoing monitoring of SUSARs. Data monitoring committees have a partnership that's very much partnering up with all of these other oversight bodies. But a distinguishing characteristic that we've always said about DMCs is they have sole access to the emerging data in an unblinded manner. Why is that? As we look at the mission of a data monitoring committee, I think of it as, first and foremost, safeguarding the interests of participants, but also enhancing trial integrity and credibility. I call it individual ethics and collective ethics. To achieve this mission, there are certain procedures that are key. One is maintaining confidentiality to reduce prejudgment. Another is to obtain unbiased judgment through having a well-informed and independent committee. I think this motivates fundamental principles then for data monitoring committees. For independent and unbiased judgment, they should be multidisciplinary, they should be independent. For reducing risks of breaches of confidentiality, they should have sole access. To provide some additional motivation for this sole access, there are numerous examples from our experience. One recent example to point this out is the LIGHT trial, which was looking at naltrexone and bupropion recently, called Contrave. And it was in obese, overweight subjects with cardiovascular risk factors. The objective of this was a cardiovascular safety trial to see if we could rule out an excess of a 40% increase in MACE, cardiovascular death, stroke, and MI. But the trial was also designed to allow a quarter of the way into the trial, after 90 events, the ability to see if we could rule out a larger margin, and if so, that would provide the ability for earlier marketing approval. Around the time this study was started, FDA had a Part 15 open public hearing on August 11, 2014, to talk about how we proceed in such studies. And there was strong consensus to the importance of proceeding in this fashion. There was also consensus among academia, industry, and regulatory authorities that if you in fact were releasing the data at this early point for regulatory approval, that it was important to have proper firewalls, that you were releasing it to those members of the sponsor who had regulatory reporting responsibility. They were firewalled away from the study team. They were firewalled away from the rest of the sponsor. And when the FDA did their summary basis for approval, they would be very sure to simply point out that the data had ruled out the larger margin, but to keep, in essence, equipoise for the trial to continue. So in essence, what happened in this study? That first analysis was done in November of 2013. The data, in fact, provided considerable evidence that Contrave was ruling out a doubling. The monitoring committee said, release the data to FDA per the data access plan. The study continued. The FDA did, in fact, approve in 2014. It was 15 months later when we met again with the second quadrant. When the second quadrant was available, the data monitoring committee recognized that the second quadrant was very inconsistent with the first quadrant. You had, instead of having 22 fewer death strokes, MIs, you had 17 in excess. But the totality of the data were still consistent with ruling out a 1.4 margin. The DMC said, continue the trial. But we said, thank goodness we're following a process where these interim data are not widely disseminated. Well, the day after this, in fact, it was later the day of this day, the data monitoring committee and the steering committee, and probably the FDA, was shocked when we realized that the sponsor had actually released the first quadrant in a patent filing. The steering committee realized a few days later that this breach had significant impact on the integrity of the trial, that they recommended trial termination. The data were then reported a year later in JAMA in 2013. There were 64% of the total planned events were in hand. There were key insights here, and the first is the unreliability of interim data. It's very apparent that these interim data were very inconsistent with what the final or more complete data showed. And the second is that breaches in confidentiality provide the potential for dissemination of misleading results and risks for irreversible compromising the integrity of trials. These interim data certainly were very misleading, and their release did, in fact, compromise the ability to successfully complete the trial. There has been considerable consensus. The Damocles group, through an extensive discussion and research into DMC best practices, concluded that there's near unanimity that the interim data and deliberations of a DMC should be absolutely confidential, and breaches of confidentiality should be treated extremely seriously. And there are formal statements of concordance with NIH, WHO, EMA, and FDA. So, in fact, however, sponsors will need to have access to interim data on a need-to-know basis. So medical monitors certainly do have a need-to-know for their Category A and B continuous monitoring. For Category C, where you would have an SAC that in fact is going to have access to unblinded data that would include ongoing trials, does that in fact need to be external to the sponsor? There's a key principle, and that is the sponsor's insights from such access to unblinded data from ongoing trials should only be shared with DMCs, regulators, and others who also have that need to know to address individual and collective ethics. And so as we're looking at these important operating procedures, it leads to another, this principle leads to another insight here. That is, those with access to unblinded aggregate data from ongoing clinical trials should be firewalled away from the study team. Is that, in fact, going to be effective? Can we effectively firewall, especially in a small sponsor? It motivates limiting access to unblinded data, wide access in ongoing trials to SRC members who are external to the sponsor, which brings an interesting thought. If collectively the SAC would have access to unblinded data from completed and ongoing trials, could there or should there be a subgroup of that SAC that's independent of the sponsor that would get the access to ongoing trial data that are unblinded? And in fact, might the DMC be one option for actually carrying out that SAC function? Remember, we had talked about principles that guide the integrity of the DMC process. Aren't these principles also relevant to the integrity of the SAC process? So in particular, for the Category C event monitoring being done, should it be done by a DMC-like independent entity, where, in essence, to have unbiased judgment as you're assessing the potential causal relationship, shouldn't you have multidisciplinary and independent folks doing this? And it's important to maintain confidentiality to avoid free judgment. A DMC-like review process to carry out this event, this Category C event monitoring, also has certain advantages. A DMC sees everything. So if a DMC is trying to understand whether or not stroke or MIs are SUSARs, the DMC is going to see TIAs, angina, and other issues as well that are additively informative. The DMC also sees the results by randomized grouping. So they're not solely dependent upon an investigator assessment of relatedness. Many data monitoring committees that I've been on, in the placebo arm, I see related events. The investigator didn't know they were placebo. Or I'll see 20 against 2, and none of them are called related because the investigator didn't understand. So by a DMC-like process, you're going to increase the sensitivity and specificity of assessing SUSARs. Well, people say, well, wait a minute, but a DMC-like process doesn't meet in continual time. Well, I do want to meet in continual time for Category AB, but for Category C, isn't periodic monitoring adequate? I mean, Peter was already pointing out correctly that there are multiplicity issues that have to be taken into account. Mary was pointing out how even at the end of a trial, you often don't have sufficient data. We saw from the LIGHT trial how early data could be very misleading. And so continuous monitoring isn't probably necessary for Category C. The data monitoring committee doesn't need to be, but could be that entity that provides the Category C monitoring. Most DMCs monitor single registrational trials. But there are DMCs, I'm on 3 DMCs right now, that are monitoring an overall clinical program that's evaluating a single investigational agent. We are looking at all of them. This would be a particularly ideal setting for a DMC to be carrying out that part of the SAC function that's looking at unblinded data. So what are some issues that have to be addressed? Salim was talking about this already too. What are the standards that we use on a data monitoring committee to take action? We look at the entirety of the data in a benefit to risk context. So if I was looking at a platelet inhibitor and MACE was the primary endpoint, and we were seeing on the DMC an increase in hemorrhagic stroke, but we were seeing fewer ischemic strokes and cardiovascular deaths and MIs, we're going to continue that trial. We're not going to report results out. And so the DMC should proceed in that fashion. If the DMC is serving in the dual role, there may be a different set of thresholds that we would use for IND safety reporting, and that's an issue that will be discussed later this afternoon. There will need to be proper communications between the SAC, the DMC, and the FDA. And in my closing slide, this leads to another important operating characteristic. If the SAC is going to have access to unblinded data from ongoing trials, it's imperative that I'm noting at the bottom that there's a charter that ensures that we're going to not only obtain reliable and timely reporting of SUSARs, but that we recognize the integral importance of maintaining confidentiality to protect the integrity of those ongoing studies.
Thank you, Tom. That was, both the presentations were really excellent and set up our discussion very well. So we have the several reactants, and we've talked previously about some discussion questions, and we're going to, in the interest of time, weave those questions in to the reactants. But I want to hear from everybody before we come back to the presenters. So I'd like to start with Amit. If you could give us a reaction to what the presenters have said, and also because of your of ACI Clinical working with different sponsors and setting up DMCs and so forth, we're particularly interested in hearing from you what existing frameworks you see for safeguarding safety already in place, whether there's consistency across companies, and what it would mean to now set up these SACs or SRCs, and could similar procedures be used? And you're mic'd, so you can sit if you want.
Yeah, fine.
Okay.
Thank you, Lisa. Thanks for the organizers for this very important meeting. I think everybody is waiting to get to the next step with this guidance from draft to the finalizations in some form. So again, as Lisa mentioned, that I work for ACI Clinical, who manages and supports safety committees, whether it's a DMC or it's adjudication. And of course, when the guidance came up, that's of importance to us to see how can we use the current technology and the procedures that we have for the safety committees into the SSE area. So from that angle, we talked to several of our clients to see where the thinking process is and how can we work together. And we talked about piloting some of them. And so what I share here is sort of what we have heard from different of our clients, small and big, pharma and biotech. And many of the discussions, I think, especially with respect to DMC, are actually very aligned with what Dr. Fleming said in some sense, because, and we will talk about that. I think that the sponsors feel strongly about the independence of the safety monitoring here as part of the SSE. And the reason, again, since morning we have been talking about the unintended consequence of study integrity being lost, or some way we have less confidence in this study given all the several times that we have gone through this unblinding and so on and so forth. So I think having some firewall or even totally independent safety monitoring, which is a similar DMC type, is very useful. And the other thing is that about safety, this is about safety, and a lot of time just looking at the DMC, even there are physicians who focus on safety, but they do also focus on the benefit risk. And so there might be a different set of physicians who might be more focused on safety, but that's something we can decide, we can think about in future. SSCs, the expectation is it will meet more frequently, not unlike DMCs, which probably are a little less frequent, 3 months in general, sometimes it goes for 6 months. So how can we incorporate that frequency into this if we go to an independent DMC-like? Because of course, no one wants to add more cost and resource, especially for small companies. It's a burden to institute another SSC in addition to DMC, for example. Background rate has been in discussions everywhere. What is the background rate? And we have talked about already many times today, and we will be discussing more. And and especially in rare disease, oncology, or older populations, there are so many studies, and is RCT, whether we should look at RWE, as we heard about in the morning, FAIR database. I heard that so many times, FDA has the safety database. Can we use, get some guidance from there? So I think there is expectation that maybe from FDA, based on my discussions, is maybe there could be a library of the safety events, the rates, maybe interval, not an estimate, for all of us to sort of abide by and say that's the FDA's expectation. And when we support and we submit SAE or SUSA to FDA, that will be checked against them. So maybe we should have uniform guidance from regulatory agency about the potential safety events rate. So I think that's then the all discussion about threshold, whether it's a quantitative discussion or it's a medical discussion, I think is both. So I think those are the things we heard from the different clients that we talked about. And some of the concerns were, of course, the aggregation, because some of if we have a program level SSE or DMC-like activity, there could be a potential for unblinding some other ongoing trials. And how can we institutionalize that when there are several trials ongoing and some of them needs to be unblinded because we are looking into aggregated safety information. And looking outside of the similar competitor drugs, who decides what the non-inferiority margin should be? So there are several of these technical questions being asked. And then the adjudication. Some of the safety events could be adjudicated. That might take time. And as we know that in DMCs, we sometimes come back at the next DMC meeting to talk about adjudicated events, which were investigator reported in the previous DMC. So how can we accommodate that type of issues in a SSE format? And so I think there are a lot of questions, a lot of interest from the different clients that we have. And we have discussed with some of them about implementation of some of these. But the question is, everybody is waiting for a next version from the draft, because everyone realizes it's not final yet. It will be changed in some form or the other. There might be a guidance. So I think those are the things. We have some implementation ideas about incorporating blinded review and then using a DMC with a different scope to do the SSC activities. And so those type of two-steps approach, we thought also that might help in reducing the issue or addressing the issue of the global alignment of the safety reporting as well. So I will stop here. And
all right, thank you for those thoughts. When is that guidance coming out, Bob? No, just kidding. I can ask that because I was at FDA working on it. I'd like to go to Erin on the phone if we've got her. And Erin, what I'd love for you to do is just give general reactions to the presentations, but also in your role working on cardiovascular trials, if you could talk about what it might mean if you had a data monitoring committee and a safety advisory committee on the same study. What do you see might be some of the challenges in operating with both of those groups and the communications and so forth? If you don't mind taking that question.
Of course. Thank you so much for the invitation to participate from FDA and Duke. So first, apologies for not being there in person. I was pretty skeptical about whether the storm would amount to much, and it turns out we got more than a foot of snow, and I have no power at home, and most flights were canceled. So let's see. So as you heard, I am an investigator with the TIMI study group at the Brigham and Women's Hospital, and as an academic research organization, we have fairly large cardiovascular outcomes trials, mostly phase 3 and phase 4, and so what I figured I could do is speak a little bit on our experience and maybe then build off of that on how there might be some room for alteration or improvement to kind of meet the FDA's goals. So we in our trials, we universally have DMCs, and with those DMCs, they have several different functions, as Dr. Fleming outlined. So first, obviously they're reviewing efficacy, and to Dr. Granger's point, this is a very targeted endpoint that have been sort of predetermined, are adjudicated, are the database is sort of screened and scoured for these events, so that there is not underreporting, and I think that's a very important point that unless you really target these specific endpoints, there will be underreporting, because we've had experiences in our trials where we call out certain events and we don't call out other ones of interest, and you just see that with the population you're seeing much lower rates than you might expect. But obviously the DMC is looking at those at periodic points throughout the trial in an unblinded fashion. But then also, we call out safety events, and one's adverse events of special interest, which again are targeted based on preclinical data, mechanism of action, theoretic concerns, prior clinical data, maybe post-marketing signals, depending on whether it's what level the trial is at. And again, these are targeted and ideally and hopefully, sort of well reported by investigators. And the DMC in our studies is looking at this at very regular intervals, depending on the study, maybe every 6 months. But for many of our trials, they're also, and so that would sort of highlight that the category C type events to some extent. The sponsors then looking for the rare events, the A and B events, and trying to unblinding as necessary for those. But the DMC for our studies also has, I think Dr. Fleming may have mentioned this, but also in our studies we'll have a function where they get a listing of serious adverse events and by unblinded at these regular safety looks. The challenge there, I think there are some challenges there. So it's obviously trying to get to the bottom of, are there any Category C events that are concerning, and is there an emerging signal that's concerning? But I think there's also some challenges with this, and I think first thing is what was previously discussed about the terms and the coding, and the fact that you have this listing of interim data, so not perfectly clean, but where there may be overlapping terms that individual are not constituting a signal. There's no imbalance, but then when you combine them, in fact, appropriately combine them, there may be a signal. So it's sort of being able to sort that out. I think, some of the other pieces in terms of having the DMC specifically be involved in IND safety reporting, which is not generally what we're asking them to do. These are statisticians and clinicians, and they may not really have the expertise that is, and to sort of know what the recommendations are and the guidance for safety reporting. And also, we've also discussed the fact that there are different thresholds potentially for, an IND report versus trial stoppage, and that's important for whoever is reviewing these events to be able to appreciate that there should probably be different thresholds for these. And plus, the DMC is obviously not necessarily going to have the knowledge of the other studies, may but may not, the other studies that have gone before. And may, although they of course know the drug that's being studied, they may not have detailed information about the sort of mechanism of action in a way that the sponsor would have for being able to sort of parse out in this listing of AEs, which ones are potentially really important and should be focused on. And then finally, again, in many of our studies, these are cardiologists, and so they may not have an area of expertise, say, if it's a pulmonary signal. So as I was reflecting on this, I was thinking about, I do think it's probably within the purview of the DMC to be able to do this, because I do have concerns like other folks do about having large-scale unblinding that is under the umbrella of the sponsor in terms of trial integrity. But I think that there's probably ways that we could do it more efficiently. So maybe, for example, having somebody, as we have, a combination multidisciplinary on the DMC already, clinicians, statisticians, maybe putting somebody with some safety expertise on the DMC who is, sort of firewalled, who could remind the group about what the expectations would be for IND reporting. I also think we have open sessions where we speak to the DMC beforehand to say, here's what's happening in the trial, here's some things of note, and maybe we may tell them about if there's been some important IND reports. But maybe formalizing a way in which the sponsor's safety group could speak to the DMC in an open session and say, this is what we're seeing. We have concerns that maybe our background rate of X, Y, and Z is a little bit higher than it should be. And so could you, while you're reviewing the unblinded data, focus on that? Or we have just, released an IND safety report on this, maybe keep an eye on other related events. And then also empowering the DMC to be able to get additional information, around, for example, causality. So going back and getting a narrative and being able to sort of appreciate whether there is, potential causality here. And then also to be able to request experience, for example, what I've mentioned before, maybe a pulmonologist for something where there's a pulmonary signal. So I do think, as Dr. Fleming was suggesting, that it may be possible to have a DMC do this. And I think in some sense they already do within, the trials that I've been involved in. But I think there's probably definitely room for improvement and for being able to sort of really target the IND reporting in the way that the FDA would like to.
Okay, thank you very much. You made some excellent points about the DMC taking on the role of the SAC, and we'll come back to this as well. Alyssa, I wonder if you, coming from BioMarin, I wonder if you could talk from a smaller company. I don't know how we define small, I agree with Janet, but smaller perhaps than a large pharma company, about the challenges in general for interim unblinding and what it would mean to set up an SAC. Could you do one in-house? Do you have enough size, basically, to put an internal firewall in, or whatever else you've been talking about?
Thank you. So BioMarin is a smallish company. I'm not sure the definition, and we do tend to run small studies in the orphan disease space. And because even the registrational pivotal studies are open label and small in size, we do try to limit the data to the appropriate personnel. So we have had some success in using data analysis plans to blind the majority of the study team to sort of the pivotal data and in the majority of folks within the organization. But having a safety assessment committee, I think, does pose a challenge in terms of manpower and actual company size, and how to sort of ensure independence and firewall of people if we were able to pull this off, and keeping that sort of protected within one group or one set of individuals within an organization. So I would agree with Dr. Fleming and with Aaron in that I think this is a role that the DMC could take on. I know in my experience at BioMarin, in our open sessions of the DMC, myself as the pharmacovigilance person on the program, that I do provide not only a review of the safety and adverse events, but also any interim events that may not be in their data cut packet. So I think it is challenging to sort of have both committees sort of running simultaneously within a small company. And I think more so it's a practical manpower issue, given that within a small company, the biostats group, for example, may all sit within the same area. So while there's SOPs and training and deviation plans that may be put in place, it's still challenging to walk by and somebody's screen or there's something on the printer. So maintaining that level of independence and firewall, I think, certainly is a challenge in a smaller organization.
Thank you. That just reminds me of experiences working. I worked briefly in a small company, and there were times I wanted to come to work with a paper bag over my head so that nobody could see my expression having looked at the data.
Exactly.
So most of you know this, but with Tom and Dave, we have two of the most world-renowned and respected authorities on data monitoring committees. Their function, most experienced. Both of these gentlemen paved the way for data monitoring committees in the NIH model and then carried that to the pharma trial model, which happened, I think, a little bit later. That's my memory. And in fact, the first DMC I ever reported to for a sponsor was in the 1990s, and Dave was running the DMC, and I learned a lot. They've written extensively about this. So we've heard from Tom. We have an opportunity now to hear from Dave. And Dave, I have 2 questions I want you to answer. So first, from your experience, what is the feasibility of a DMC taking on the IND safety reporting role in terms of dealing with different thresholds, different types of risk assessment versus benefit risk assessment, and so forth? And then second, if a separate SAC was set up and you were on the DMC, what advice would you have about these 2 groups functioning together, recognizing they may come up with different advice about the same risk or same signal and so forth, just based on your extensive experience, if you don't mind?
Yeah. Well, I attended my first DSMB meeting 45 years ago this month. I didn't have any gray hair in those days. I guess that's an association. I don't know if it's causal or not, but in any case. And I think I'm old enough, I'm going to answer your questions, but I want to add a few of my own comments.
Absolutely.
So I want to put in a plug for the paper that, in some sense, Janet Wood has summarized. It's not an easy read, but it's a very great read because it probably better than anywhere else, it summarizes what's going on. And there's a nice section in there about the state of affairs about the data we collect. None of this matters if we can't get the data straight, in my opinion. And I don't think we're getting the data straight. It's, as Chris Granger alluded to, we have a passive system. And from this passive system, we try to get structured data, which is not possible. So Celine was complaining, as I have been for many years, if we want good data, then structure it. We know what the deal breakers are. We're talking about events that would stop a trial in its tracks or a product in its tracks. We're not that stupid. We know what they are, most of them. And of course, it depends on the context of the disease. So let's collect that data in a structured way. If you want to collect all that other stuff, fine. But there's 2 ways to hide a signal. You make it so granular you can't see anything, or you drown it with noise. And that's exactly what we do every day. And I see page after page of adverse event reports. I have no idea what it means. It's full of 1s and 0s and 2s. You can't make any sense out of that. Anyway, so that's problem 1. I am amazed when people say, I want you to be on a DMC, but you're only going to look at safety. Really? It's always a risk-benefit assessment. As been alluded several times, there's many cases where, yeah, you see some problems, but the benefits overwhelm the problems, and you're willing to put up with a little bad stuff if you got a lot of great stuff. So I'm always amazed, and I refuse to participate in such studies, by the way. I don't want to get caught in that. I have been puzzled from the beginning of these guidance. If you have all, I call it the XYZ problem. If X is the data you know about, it's published, it's been presented, and now you add Y, and you get X plus Y equals Z, and Z is known. I think my 4 grandsons could solve that equation and figure out what Y is, as you've unblinded the ongoing studies. So I don't get it. If we're trying to protect the blind, we've set up a perfect system to unblind it. So I'm puzzled by that and frustrated by that. Firewalls. I don't believe in firewalls. I agree with you, firewalls in a small company would be asking the impossible. It's just asking too much. But even for large studies or large companies, whatever that means, I still don't believe in firewalls because it's not that anybody's handing you a piece of paper with the data. It's your activity, your mood, the amount of time you spend in meetings. In my groups, when I was at the NIH and at Wisconsin, I always knew what was going on, more or less, because I watched the activity. I could tell, whether they were smiling or not smiling. So I don't believe in a firewall, big or large. I would support the concept, I'm calling it DMC plus. There are examples. The AIDS group, which I was a part of along with Tom, we monitored God knows how many, maybe 50 trials in that portfolio. It was very structured and it was very demanding, but we did it. Another sponsor I've been working with for 10 years, we monitor all their trials for this indication. So it requires a little more work, more discipline, a little more education, but you can do it. I don't see how you could have an SAC that didn't have the principles, the operating principles of a good DMC, I think, which what Tom was mentioning. If they were not behaving that way, I wouldn't know what their value was. So, you can call them what you want, but if you have monitoring one study, one monitoring company for one study, maybe you need a Uber DMC Plus to look at the big picture. But many DMCs have and can monitor a portfolio. So I think it's possible to do that. In fact, it's been done and done pretty darn well in my experience. But, again, at my age, I've learned to never say never. So there's always the exception, perhaps. Thank you.
So I think we heard Dave's grandsons are eligible for DMC service. Is that take home? All right. This is a serious topic. So, Matt, the FDA always gets the last word.
Get the last word.
So I know from my stint at the FDA, I can't say we anymore, used to we, get really nervous that there's too much unblinding going on. And Tom gave a great example of exactly that happening, or our concern about it happening. We were so nervous about it, we had a Part 15 meeting on interim unblinding in 2014 that Tom presented at. What are your reactions to what you've heard, and what are the concerns from a statistician in the safety group at CEDAR about setting up SACs, having interim unblinding, possibly some amount of that happening inside the sponsor and so forth?
Well, thank you. How I'm going to approach this is I kind of wanted to draw a parallel to how we evaluate cardiovascular risk in the type 2 diabetes setting, where, if you're not familiar with that 2008 guidance, is ultimately it sets up a framework where you have to rule out a 1.8 relative risk of cardiovascular risk at stage 1, which we usually use for gaining marketing approval, and then later you have to rule out 1.3. And one approach to address those two risk margins is sponsors have done a single cardiovascular outcomes trial where you do an interim look at that in that ongoing trial to look at the 1.8 risk margin. So this setting very much sets up, I think, something parallel to how an SAC might be thought of, where a sponsor has to be unblinded to those interim results. We saw this early on. We saw it as a challenge. Alyssa mentioned we had a Part 15 meeting to kind of look at, get other feedback, but also to kind of evaluate how we were doing things and what we put, the processes we requested from sponsors to hopefully have some degree of a firewall of that data for ongoing trials. So really what we do in that setting, or what we've requested, is we have sponsors fully document who's going to have access to that interim data, what their role is in the company, what level of access they have to the data. We also ask for information on how the data flows to that internal group within the company to ultimately, they have to give us the information from that interim cut of the trial. So I think what we've learned, after implementing this and using this approach for several years, is that ultimately on paper, it looks good. It's the firewall. We think it's there. It's a small group. We feel everything's in place. They have confidential agreements. The data flow seems to be correct. But I think in reality, there really is no true safeguard to say that that firewall does exist. And Dave brought up a great point, is there could be a mood within the company. There could be something going on. So for me, the question is, trying to parallel this into the SAC world, is if you have internal sponsors looking at this interim data, are they ever truly firewalled? Can we believe it? And where it's hard is, as regulatory, is we try to figure out, did that firewall hold or not? It's really hard to do. We're not there watching the trial ongoing. We're not sitting in the company. We don't know, and we probably never will know. And when things do not happen, Tom brought up a great point, the LIGHT trial, which our division reviewed, and it was only after a little tip here, a little thing there, things just didn't look right, that we started really asking more questions, and that's when we figured out, that firewall isn't in place at all. So maybe it was luck, we don't know if we would catch something like that again. So it's very difficult to really do. Another point I wanted to bring up is when we're looking at interim findings from ongoing trials, and again, most of my experience is going to come from a lot of the post-marketing setting. We're looking at large outcomes trials. They're multiple years in duration. They're big trials. When we have, and again, these I think are the ideal trials as brought up earlier, that if you're going to find something for safety, these are probably the right ones because, you know, these big ticket safety items, they're not at a high frequency rate. So you do kind of need this large trial to look at it. So when we're in that setting, and we have been in a position where we've actually had a DMC come to us and say, we have a signal, we have something we're concerned about, and what we've talked about internally at FDA is, well, what are we going to do with this? We have one side of the equation, or we don't have the full picture. You're going to maybe only report to us a risk. So in those situations, we have asked to say, okay, report to us. Don't tell us any, what direction that signal is, where it's going. We just want to know, okay, you found a signal. What are your plans? And we deferred to the DMC to ultimately protect the trial and the patients under their original purviews. So in general, I think where I would come down to things is when we're looking at this category C, is I would probably lean towards where Tom's proposal is that ultimately for these types of events, the DMC is already situated to really look at them. And if for some reason a sponsor does need access to the interim, of any interim finding, that it's limited to as few as possible. And if there's any way to kind of roll them off as the product's maturing, I would feel that's an ideal situation.
Thank you so much for that insight. So I want to ask first if Tom or AJ have anything to add, having heard the reactants, before I go back to a couple more questions.
So thank you, Lisa. I guess this has been a great discussion, and I sort of took some notes here, and I'm looking at four major themes, and one is external versus internal. I think we've all discussed that, and clearly, I think everybody agrees, if we have external bodies, the firewalls will be more effective. I don't think anybody disagrees with that. The issue then becomes, what are the logistical practicalities of that if we have SACs across the board for the developmental portfolio? I mean, I think it's an issue. So having them all external is one way, or having an IDMC take them on, but then we would have to have some way to look at studies that don't have IDMC. So we would only be restricted looking at category C for the large pivotal studies. We would not be able to look at it for the small, and that's fine. I think that's one way to go. But the remit, obviously, of the IDMC versus the SAC would be the second point, which is a bit different right now, because IDMC is looking at benefit risk, whereas for the SAC, presumably, we want to identify early signals, early emerging signals that are not showstoppers, that are not going to stop the study, that may not even impact the protocol conduct, but that we think are potentially causally related. That's a pretty different bar. And so that, I think, is going to have to require a major updating of all the charters, and the DSCMC members are going to have to realize that, look, we're going to have to now understand that any emerging signal would require us to identify the sponsor and perhaps the FDA. Which brings me, I think, the third point that's been discussed sort of holistically here is interpretation of interim data. I mean, that is an issue. I mean, if we're going to look at interim safety data with small numbers, and we all realize that the contemporaneous control is the best control, there's going to be a lot of instability, right? You may see a 3 versus 0 at 3 months of a really important event, and at 6 months, it may be 4 versus 3 the other way. So where do you draw the line on sending that in? And that, I think, is an issue that will need some further clarity. And then finally, if there is an SAC constituted in a study that has a DMC, I think it may be even somewhat more complicated. So to take your point, for the type 2 diabetes studies, we would probably have a IDMC, a adjudication committee for the cardiac endpoints, and we may well have a safety endpoint adjudication committee for pancreatitis. And now we're going to have an SAC on top of 3 other committees. How do they interact? And I must confess, it would be very difficult for a sponsor to question. I wouldn't want to question if a body that's looking at unblinded data says you've got a signal. I have any way to put any judgment on that because I have blinded data. So now we may have 4 committees. And would the SSA then talk to the adjudication board? I mean, there are a lot of complications there. So.
No, I think those are very good points. Thanks. And Tom, your reactions too. And if I can, I'm going to embed another question. So we point out in our FDA guidance, and Bob and I have had many hours of conversations about what trials need DMCs, and the fact that since the FDA guidance was published in 2006, many, many more trials now have DMCs than did at that time. But there are trials that don't have DMCs. And so we haven't talked, I don't think anyone has, about a trial that doesn't have a DMC, but then would have an SAC. What do you see are the challenges there in terms of, with the SAC in place, would there be a temptation for the sponsor to expect the SAC to maybe all of a sudden now act like a DMC? I mean, do you think that they could keep their remit focused just on IND safety, or do you think there'd be a concern that it might morph into a DMC? Just if you have time to answer that.
So I'll try to circle to that at the end, but I'll try to also be brief. Listening to the thoughtful comments by others, a really critical issue here is we do want to be able to identify in a timely way safety risks. Everything is in the context also, though, of benefit to risk. And we obtain so much valuable insight in evidence-based medicine from our larger randomized clinical trials. And we surely cannot compromise the integrity of those studies. And confidentiality is integral to the integrity of those studies. So there's no absolute. There are reasons to, in fact, share confidential information beyond the DMC. I call it need to know. But we should be persuaded that the risks that will be induced from certain levels of sharing of that information is kept as small as possible, and that it's unavoidable relative to the gains that we hope to achieve. So my sense is, for category A and B, yes, we do need to have medical monitors. Practically speaking, they're going to be within the sponsor. They are going to have continuous monitoring. They are going to, in fact, in some cases, have access to unblinded data. And my sense is the gains that we get from that offset or greater than the level of compromising integrity of the studies. I remember in 2008, when we were on the Endocrinologic and Metabolic Drugs Advisory Committee, and we had been in type 2 diabetes, and we had seen so many studies done that had 300 person years involvement, and we found out non-inferiority on hemoglobin A1C. And we were saying, we do not understand adequately what the effects are on microvascular and macrovascular complications that will come from large-scale randomized trials. But we were practical, saying we wanted a paradigm shift from 300-person studies to 40,000-person-year studies, which is what it would take to rule out 1.3. And so we took a compromise, and FDA was very responsive in implementing this compromise of the 1.3, 1.8, where you get into the market at 1.8 and you finish it at 1.3. So in essence, it was a compromise to move us from 300 to 40,000 in a feasible, practical way that still got these drugs onto the market in 8,000. But it's imperative that we get those gains with the minimal loss of integrity to those studies. And FDA and industry and academia has worked very hard to think of how to do this. And it does depend to an extent on firewalls. And I gave an example of a failed case. But I will say there have been many successful cases where sponsors have carried this out. Is there a better way? I would love to not have to firewall it, but I would love to not have to release the data when I'm on a data monitoring committee and I know that the study truly has to rule out 1.3 to be reliable. And yet, if we're going to be able to have a feasible approach and we're going to get timely access to interventions, this is what we have to do. Well, now we're talking about Category C events. And for Category C events, to me, it's imperative, as has been pointed out, that you need to understand these in the context of randomized comparisons that are unblinded and that are based on understanding the totality of the data. And that leads to me to the conclusion that it's a DMC-like, doesn't have to be a DMC, but it's a DMC-like process. The DMC may be ideally suited to do it. But if it's done outside the DMC, I have great concerns if it's done under the umbrella of the sponsor. It would be ideally separate. And it might be coexisting committees where the motivation for this could be that the SAC is looking at many studies, although a DMC could also be looking at many studies. The fundamental issue here is I'm not persuaded that the gains that we get by running an SAC within the sponsor, particularly in a small organization, broadly unblinded, are offsetting the risks to the integrity of that research that's integral in an evidence-based medicine. And this is the trade-off. So do we need DMCs in all studies? No, we don't. And there is clearly an understanding of settings where you do, where it's irreversible morbidity, mortality, or where there are major risks for the intervention. There are, however, a lot of other settings where a DMC can be very, very helpful to enhancing the integrity of the research. When we're on monitoring committees, my first and foremost responsibility is safeguarding patient interests. But it's also to help enhance the integrity and credibility of the trial, which also is improving quality of study conduct. And it's also based on, in settings where sponsors now, and it's happening more often, have said, we want you to monitor all of the development. In fact, it was something I think Bob Temple said to me a long time ago, which is, shouldn't a DMC potentially monitor a program of related studies? And as Dave pointed out, we effectively monitored 50 trials. We monitored on the HIV/AIDS data monitoring committee all of the ACTG CPCRA trials for 20 years, which was an extremely efficient way for an independent entity to be able to understand the totality of the data. So my sense is there's no single right answer for where you do have one, but when you don't have one, then I certainly do see that an SAC type arrangement could be very useful, but it should be as independent as possible. And I agree with Dave, if you're going to monitor safety issues, you need to understand them in the context of benefit to risk. And so you should be seeing totality of the data, which, by the way, seems to me that I'm circling back to saying data monitoring committees are really key.
Okay, so we have 10 minutes for, first of all, session 2 needs to get some credit for getting us back on time. We have 10 minutes for audience speakers before lunch, and if you would go to the mic, while you're going to the mic, if we have any, audience questions. If you have any questions for this terrific panel that's up here. We did get a question on the phone from Waldhoffen at AbbVie about why can't a DMC serve the purpose of the SAC in terms of determining when to report SAEs to the FDA, and I think we've talked about that quite a bit already. But then Walt went one step further and said, could the regulators develop rules like setting p-values for these imbalances and so forth? And so I'll just let Matt answer that, and I'm guessing it's a one-word answer.
No.
But in all seriousness, we haven't talked much about thresholds. Tom mentioned them in his presentation, but that hasn't been one of our topics. Maybe it'll get discussed more this afternoon. But that an imbalance to one group may not be an imbalance to another group, and I think that gets into the DMC SAC as well. Yes, thank you.
Valentine Hill from Novartis. I have a question. Since this morning we are talking about aggregated data. And I understand that the DMC really, when I am looking at one single trial and I am looking at a DMC for this particular study, I am looking at the clinical database. And I'm really looking at the totality of the data, as you just mentioned. When we are talking here about aggregated report for this particular activity, are we talking about pooling database, which are clinical database, or are we talking about looking at our Argus system and looking only at the serious adverse event, which can be quite different if you want to understand the totality of the risk? So the question is really, clinical database pooling versus really only looking at Argus, which is really only the SAE systems.
If I'm understanding the question, I know this afternoon there's going to be a discussion about aggregated data and meta-analysis. The setting that I've been talking about, I'm on several data monitoring committees that are monitoring the entire clinical development program for a single experiment investigational drug. We monitor those studies as independent studies. Each study is complementary in terms of asking a certain question about the use of the intervention in a certain manner. But we do have the benefits of seeing collectively what the adverse experiences are. So a correct point that's been made is if a monitoring committee is monitoring a single trial, and yet there are many ongoing studies, that monitoring committee is very well informed about benefit to risk in that trial, but doesn't see other studies. Well, the sponsors that have set up single monitoring committees to monitor a program, I think, get the best of both worlds because it's an efficient process for each study to be monitored for its independent objectives, but where the DMC does have the insight collectively across the program of the emerging safety experience.
But when you are looking at, in this particular experience, you are looking, for example, at the vital signs, at the laboratory data. You are looking not only at SAEs, right?
And
it's a broader picture that you are looking at.
Amit, you want to?
Yeah, I just wanted to add word from Dr. Fleming that we believe that if we have to have SSC sort of incorporated within the DMC plus type of situations, it would be good to have a program level DMC because of the reasons we already talked about. So it will go back to the clinical database. That's not for that trial, but for other trials of the same program as well.
Yes, next question.
I agree that for reporting, we need to do an unblinded analysis. Somebody needs to do an unblinded analysis. But, and I don't know if this is going to be talked about in the afternoon session, I think if we take a two-staged approach, that we could leverage the scientific expertise and the medical judgment of the multidisciplinary safety management team. That's where the strength is in the study team, the blinded team. So there are methods that, quantitative methods that we can use to look at the blinded data if we have information, again, looking for the wolves, not the chipmunks. And then if there's some discomfort with what is being observed, then you could reach out to an unblinded group. And in that case, if it's not a broad and frequent unblinding, like for an SAC, which is recommended in the 2015 guidance, then we could take advantage of this two-stage approach. I don't know what kind of reactions the panel would have about that.
So,
AJ.
I guess in some ways we do that now. So I could give you an example. Now, this may not be ideal, but there are several instances where the SRT, which is blinded, will look at a cluster of events, and some of them may be unblinded because they were SUSARs. Others are not unblinded. Hypothetically, let's say we see a serious event, and we have a total of 20 events, and 5 are unblinded, all 5 are on active drug. Well, that makes us somewhat worried. What we would probably do is send that information to the IDMC and say, look, we've got this cluster of events. We'd like to work with you. What more information on AEs, to the point that it is a data dump, but can we facilitate getting you the appropriate AEs and lab values for you to be able to make some sort of adjudication on this cluster of events? We would work with the IDMC to come up with a data set that they could use to see if there's something concerning. Now, I think the challenging part comes in, and maybe Dr. Fleming, you could speak to this. Let's say the IDMC then says, yeah, there's an imbalance, but it's nowhere near to tell us that the study should be stopped or changed. How do you communicate that? What we would likely do is have a steering committee, which is maybe the CMO or one other person that's sort of sitting between the IDMC and the study team to get that information. I don't know if you've had similar types of.
So what I agree with you, and my sense is the monitoring committee may readily, in a case like that, say, from the perspective of equipoise in the trial, we should continue. And if I were simply monitoring the trial to safeguard patient interests and to enhance trial integrity, I wouldn't do any reporting. If, in fact, though, I needed to carry out IND safety reporting, then we'll talk this afternoon, I think, about what nature of thresholds would be in place. And if, in fact, this event required then timely reporting to FDA as an IND safety report, then that should happen, and we should talk about what's the most effective way for the Data Monitoring Committee, upon seeing this, to report this to an SAC or to the FDA.
And I don't know, Alyssa, if you wanted to respond at all about, no, actually I was looking at Alyssa, about whether that's practical, the two-stage approach he was just talking about, or maybe that already happens in BioMarin.
Yeah, I mean, I think that that's pretty common practice, and that's typically how but our trials, for the most part, as I mentioned earlier, tend to be open label. But even so, if there's even, any even small level of concern, it would be then to bring it to the DMC. And if the trial was partially blinded, partially unblinded, there may be data that is protected that the SMT or SRT can't see, and then you could bring that unblinded data to the DMC for review in a closed session.
Right. So
I'm sorry. There's a lineup behind you. And I promise to finish on time.
Hi.
Okay, Bob.
I just want to separate the question that's fundamental to our guidance and the larger question of whether you should stop the study or something. That is not what the guidance is about. It's only to determine whether the event that's being described is really increased in the drug group. That's all it's about. It's not whether you should stop the study or anything like that. It isn't even about whether you should change what's in the informed consent, all of which is relevant. So my question is, what are we worried about potential unblinding? So I've got GI bleeds, and they're serious. And I have a bunch of them, and I want to know whether there were more in the group that got the drug and the group that didn't. So I unblind only the drug that the people who had the bleeds were on. I don't unblind anybody else in the study, just the 7 people who had bleeds, and I find out what drug they were on. Why does that make me worry about the integrity of the study? It's a tiny fraction of the total patients. We don't know anything about the effectiveness of the drug in those people, at least if they don't look at that. All they look at is what drug the people who bled were on.
Right. So Bob,
what are we worried
there's
about?
a continuum. And the example you're giving actually feels to me a little bit more like category B than category C. So I'm with you.
No, wait,
a very small number
Sorry,
of
GI
people.
bleeds are common in this population.
Well,
The presence of a GI bleed, it doesn't tell you anything. You got to know if it's
But if
unbalanced.
they are common in the population now, then we'll talk this afternoon about thresholds, but I'm not sure why 7, unless it's 7-0, is going to be sufficient for you to declare that you have causality.
I'm not saying it is. All I'm saying is that to define whether it's causal or whether you're worried about it, all you need to find out is what drug the people who bled were on. Why does that make you worried about the integrity of the study?
Because, in fact, in principle, to do this right in an informed way, in many instances, it's more than 7 people. It would be a much larger number. And in fact, if you really wanted to understand this, it probably is best to be understood in a broader context. I think the example was used about if you're seeing strokes, what do the TIA show? If you're seeing MIs, what do the angina? You're going to look at more things. And the Data Monitoring Committee could do that. So could an SAC, but as the SAC sees more data, now you've made a transition from a small number where if you unblind, it doesn't compromise integrity, to where it's now not a small number.
Let me keep going though. But you don't know anything about whether the drug worked in that population. You're not even asking that, and I would argue you shouldn't even look. What are you worried about? You now know that the people who bled or had whatever it was were more likely, 7 of them were on drugs, 7 were on placebo. What am I worried about?
If I
Yes.
apologies.
No.
I think that would, if I read it right, Dr. Temple, that sort of is like the two-step process that we were talking about before, where a team might see a cluster of events, have somebody unblind them. I think, at least my understanding of the proposal right now is for routine unblinding of all SAEs across the whole study. There, I do think the amount of unblinding is large, as compared to targeted unblinding, where you see a cluster. And I think that's where I see the bigger difference, that we're now proposing routine unblinding of all SAEs by this SAE committee.
Suppose that all they knew about the patients were what drug they were on, and they had no idea whether they had study endpoints or not. All they knew was the side effects.
Agreed, but
How worrisome is that?
It just gets to the point that if the committee is internal, there's going to be a lot of people that are going to be looking at unblinded data and concerns about maintaining firewall. That's all.
So you're worried that looking at that will reveal, in fact, to some people whether they did well or did badly?
No, it might reveal, if you start looking at number of events, there are going to be a number of people that are now unblinded.
Yeah, and Dave, you wanted to say something.
Well, you'll have this discussion this afternoon, I'm saying, but one thing you learn very quickly on a monitoring committee, 7-0, I always make a bet, it'll go away. And usually I win most of the bets. Because you realize that data, over time, wiggle and waggle. So I don't know what that 7-0 is telling you, but I don't believe it's going to tell you anything.
That's a totally different question.
No, it's
Now
not.
yes,
Why,
it is.
why do you need to know?
Look, a fair question. You're asking whether type C events ought to even be analyzed at all. It's a different question. I'm not dismissing it. I'm just saying it's a different
But
question.
I'm saying you want to know something, and I'm saying 7-0 isn't something that probably I would even pay too much attention to unless it became 20-0.
So Bob, you correctly noted.
I'm not
in
dismissing
the
that
guidance
question.
of
I'm just saying what we did.
2015, you noted that it was important for not to deluge FDA with everything that's in SAE. There needs to be evidence of causality, relationship.
That's right.
What we're saying is, when you monitor a lot of trials and you see 7 events that are common, it's not just that we would go on because benefit exceeds risk. It's you realize that that type of data is completely unreliable as to whether or not treatment is inducing that. So by your own decree, you don't want to see those data, not until the numbers are larger. And now we're talking larger numbers. Now we're talking broader unblinding.
All I'm saying is you're raising a different issue. You're saying these type C events are almost never detectable in a meaningful way, and we should forget about it. That's a different question. I'm not dismissing it. It's a different question, though.
So we
Because
have to.
the current premise was that you are supposed to look at these events and see whether they're unbalanced, and if they are, report it. Maybe that's a mistake.
Define imbalanced.
Well,
because by your own
that's the
correct
question, isn't it?
declaration, you do not want to see noise. You want to see signal.
Okay, so today is the International Day of Women.
So that means women can prevent lunch? Is that the idea?
I wanted to make sure Erin didn't have any last words. The woman on the phone, Erin, did you have anything else you wanted to add?
I just have 2 comments. I think one of the challenges with the discussion that was just happening is, again, how do you know to look at GI bleeds? If you're not looking at unblinded data, which I think is the challenge that we were discussing before about knowing what background rates are, which I think is very hard to do. And so, that's one challenge. And I think the other thing that I just wanted to maybe pose to the group, I don't know that we'll have time to discuss it, but is if you had a separate SAC from a DMC, and the idea that we, would sort of want somebody who's assessing safety to have the totality of information and to be able to see the benefit-risk ratio in general. I guess I'm a bit uncomfortable with the idea that they would be then also seeing efficacy. We normally limit the efficacy looks, and so that's just a bit of a challenge, I think, if you have another group who's also looking at the totality of the data.
Right, so we have lots of challenges, but we'll clear them all up this afternoon. Janet, I'd love to call on you, but they're really pulling the rug out from under me up here. Thank you so much to our panelists. This is a great discussion.
Okay. Okay, thanks to Lissa and the panelists. We're going to go ahead and take our lunch break. It's not a full hour now. It's going to be whatever it takes to get us to 1:30, which is when we're going to start the next session. If you're not able to finish your lunch, and again, there's a list of restaurants at our registration desk. There are a lot of restaurants in the area. You can bring your food back into this room. You can also leave your stuff in this room. We're going to be here, so it'll be fine.
Mrs. Strauss?
Yes.
Okay, so I'm about to put this mic clip on your shirt right here, just so you know.
Is everybody mic? Are we waiting for something? Where? Right now? Okay, I can invite you guys to join me up on stage. Okay, welcome back, everybody. We're going to go ahead and get started. In this next session, we're going to take a bit of a deeper dive into the approaches for determining the threshold for reporting adverse events and consider key factors that might influence over-reporting to the FDA and how potentially to the best way we can mitigate these challenges by setting an appropriate threshold. Joining me up here on stage are Ann Strauss, Associate Vice President, Clinical Safety and Risk Management at Merck, as well as Anand Chokalingam, Director, Epidemiology at Gilead
This
Sciences.
is on but
Salim
muted.
Yusuf,
Once you sit down on the stage.
who's getting Professor, Department of Medicine at McMaster University, and then Gerald Delpan, Director, Office of Surveillance and Epidemiology at FDA. This session will start with a brief presentation from Ann, and then followed by panel comments, and then we'll open it up to the audience for discussion. And if this session is like anything like the others, I'll try to start the audience discussion a little bit earlier, because usually in public events we have like one or two people will go to the mic, and we like 5 or 10 minutes of audience discussion. But for this group, we have exactly the same number of people from this morning. We didn't get people trailing off, and we usually have a lot of people lined up at the mic. So I'll make sure we leave some time for you all to ask your important questions. Ann?
Okay, so. Oh, thank you. I'm never very good with this technology. Oh, turn it off.
Okay. Just keep going.
Oh, okay, sorry. Lunch break. So good afternoon. It's a wonderful opportunity for us to present at this meeting today. As Adam said, I work at Merck in clinical safety and risk management. And we've actually had a project team that has been working on the guidance for about 2 years. That's called the Long-Term Quantitative Enablement Team, and I'm presenting on their behalf. So, as you all know, this topic is, when do you report an aggregate IND safety report? So, and then once again, and I know you all know this, but I just wanted to reemphasize it, is that the way we look at it is we would send down an aggregate report down to FDA if we thought there was a causal relationship between a drug and a serious and unexpected adverse event, or if we're seeing an increased, clinically important increase. So that does sound relatively simple, but reading through the guidance again, I think one of the points that is so important is that to be able to figure out when to send something down to FDA as an aggregate IND report is a complex judgment call. And I think one of the reasons why we all have jobs, and it's generally not a simple application of a planned statistical analysis. So we looked at that and we thought, okay, so how can we go about it? How can we have some sort of framework whereby we can meet the guidance? So I think what's important from a practical point of view, and what we at Merck do, is that for all our products, we have a safety review team. And that safety review team consists of multidisciplinary experts, and we review aggregate data on a routine and ad hoc basis. And I think, especially for aggregate reporting, we at Merck and at CSRM have had a much more intense relationship with our statistical colleagues. So the one thing is to have those experts. And then the second point to consider is that we feel it's important that to have an aggregate program level plan, so have what we call an AGSAP, so that when you have a program going into phase 3, you at the end of phase 2 in general, you plan and decide, okay, how are we going to monitor this? What are the important events that we are concerned about? What are the important anticipated events that we will be looking at, and what are the background rates? So that you have a strategic plan before you start. Because I think in the past I've been at companies where you've just really looked at the data from more of an ad hoc point of view. So this is definitely planning before you go into phase 3. So if you look at the key components of our AgSAP, the first part is, obviously looking at program level safety topics and populations of interest. Then we also have what's known as a blinded ongoing analysis of safety evaluation for blinded data. And then we also have another document where you look at unblinded data. And then obviously the fourth part of the AgSAP is we prepare for our filing. And having this all together helps with the reporting up to FDA. So just to go through, so the blinded ongoing analysis of safety, you're basically trying to evaluate the safety profile of the product, and you're trying to detect emerging safety topics. And these safety topics and signals are validated by the safety review team. And as I said at the beginning, the very important thing for the blinded ongoing analysis of safety is being able to know what anticipated events you want to look at, try and find background rates, because I think background rates are not always that easy to quantify, and it's not always that easy to, even if you have phase 2 data, it's often just a one data point in time. So, and then also quantitative frameworks. So when our statisticians and our safety physicians got together, we said, okay, what are the key questions to answer if we're looking at blinded data? Do we have more events occurring than we expected? Yes or no? And what is the magnitude of this relative risk? So we came up with a quantitative framework to stimulate safety review team discussions and improve conversations about safety monitoring. But these are not decision rules. So just to show you, this is work that was done by my statistical colleagues, specifically Greg Ball, and I'm going to read this because I'm definitely not a statistician. But basically, it's coming up with probability threshold boundaries for incident rates. So if you have an event that you are concerned about, you can see as your clinical trial goes along, you can see whether your events are actually going to be crossing the threshold that you're going to be concerned about. I mean, if you look at, obviously that red line is the bit that if you cross the red line, you're going to be concerned about. So for instance, as an example, if this is a simulation, but if we had a event that we were concerned about and the safety review team was looking at this data, we can say, okay, looks as though what we're seeing is not terribly alarming. If we had to see it going, increasing and crossing the red threshold, that is when we would, the safety review team would look at the blinded data, not unblind, but look at the blinded data and see whether something additional needed to be done. So obviously the first thing is to see whether there is an increased risk, and then the second part is to see what the magnitude of the relative risk is. So this figure can be used to evaluate the magnitude of risk evaluation under assumptions of background rates. So the x-axis is the pooled event rate at a given moment, and the y-axis shows the probability of risk evaluation above a certain threshold. So it's basically putting the data that we have into a framework where we can see, okay, this is higher than we expected, and this is the magnitude. And so yes, this is something that's concerning, but that doesn't mean we would automatically then send a report. We would then have the clinical interpretation of the data. So that's with regard to looking at blinded data. We also look at unblinded data on an ongoing basis. But the unblinded data, because as you know, a lot of our studies are actually unblinded, but we do, so they're not, there's not that they're unblinded, they're open label studies. So we look at those data when the trials are locked, and we once again try to understand the safety profile of the product. And the way we would actually look at quantifying that is by, one of the ways that we do is based on Janet Wittis's paper, looking at those rules. We've developed a Bayesian version of this type of rule that, Dr. Wittis published. So basically, the way that we approach this at Merck is that we have our RMST, which is the same as the safety review team. They are reviewing the blinded data on an ongoing basis. Once the studies are completed, they'll look at unblinded pool data and other data sources. If they saw something just from their review they may see something just from a review from a clinical point of view, or they could see something from the quantitative frameworks that trigger or that cross a threshold. The RMST will then review all the other data that's available with regard to that specific event. And if we have a DMC, we would then pose the question to the DMC about whether the DMC thinks this is causally related to the product or not. And if the DMC did feel that, they would feed it back to us, and in general, if it was actionable, so if we're going to update the informed consent or update the reference safety information or CCDS, we would send it down to FDA as an aggregate report. We don't have DMCs for all our studies. If we don't have a DMC, our chief safety officer would generally look at that data at that point. So, so sorry, I hope I didn't, that's a lot of data that I sort of like rushed, that I hopefully didn't go too fast. But the way that we're trying to do it is having the expertise, when something meets a threshold, not just having everything go down to FDA, but having clinical input with regard to that, and really seeing whether we think this is due to drug, whether it's a serious event, and whether patients need to be informed, and we then inform, at the same time we inform FDA.
Okay, great.
Oh, sorry. Conclusion slide. So, appropriate expertise, proactive strategic planning, ongoing safety review, and some sort of quantitative framework.
Okay, thanks, Barbara. So I'm going to just ask the. Anne. Oh, I'm sorry, Anne. What am I looking at?
Postprandial.
It says thanks, Barbara.
Barbara.
Okay.
I don't mind.
Let me do. Anne. Okay, thanks, Anne. So I'm going to go ahead and turn it over to our panelists for some opening or sort of kickoff reactions from the presentation, and you're welcome to stay seated if you'd like. Or okay.
Well, thanks very much, and it's a pleasure to be here. I thought I'd just add a few pieces which we've been thinking about at Gilead. Just general sort of considerations. I think, as has been alluded to earlier this morning, there's definitely a need to have a good comparator. And so one thing that we're considering when we think about thresholds is, well, when do you want to use a statistical test? I think we've discussed possibly avoiding statistical tests when the data come from really different sources. You really need a really good internal comparator where you can adjust and make appropriate comparisons. I think the other thing with regard to another point I'd like to make, so I have a number of points I'd just like to go through and ask questions even of this panel and of the audience. If you have a good unexposed comparator, you have many options to make comparisons. But you need to give consideration to things like multiple testing, especially if we're looking in the area of unanticipated SAEs. Things like multiple corrections, like the doubly robust FDR, or other corrections might be important. And then, of course, setting an appropriate p-value threshold. And I'm going to leave that one on the table for a moment. But I think when we think about the different events that we have, there are a number of different types of thresholds we can use. Do we take this descriptively? Do we look at things simply from a standpoint of looking at a rate difference between two groups, between a comparator arm and a treated arm? So we might think of, for example, for serious adverse reactions that are known, we might be looking now for frequency category shifts, something big picture. We might be looking at proportional changes or doublings when the risk is at a certain level already. We might be looking at absolute rates and absolute proportions. And then, of course, if there's a comparator, a reasonable comparator, we'd probably bring in some sort of p-value based on maybe something with an FDR calculation built into it. Those are just ideas. These are not any specific position that we've taken, but just ideas for thought around thresholds. But it's important to remember that there are a number of trials that we have at Gilead, and I'm sure others have as well, where there isn't an unexposed comparator. And so it's not really clear what to do here. I mean, coming up with a background rate is problematic, as we've discussed. But we might often have a study drug versus study drug plus best alternative therapy. So you see, we don't really have an unexposed group. We've been talking today a lot about when to unblind, how that impacts trials. But one other thing that I'm particularly interested in talking about as an epidemiologist, as it applies to background rates, is the issue of observational studies, because the guidance also makes reference to the need to summarize and synthesize data from ongoing observational studies when we have an ongoing clinical trial program. So that's something that is certainly an important consideration, and that has some concern for us when it comes down to coming up with an appropriate comparison group there as well, because in many instances we have post-authorization or post-marketing studies going on in many different countries, and these are usually often just cohorts of exposed patients with no built-in comparator. And finally, the last thing I want to talk about, and again, pose in the context of background rates, because that does seem to be a hot issue, is where claims and EMR data fit in. I think that it's been discussed today how challenging those can be. Deriving a solid comparison group based on claims and EMR data is very challenging. Inclusion-exclusion criteria, their exact event definitions, ICD versus MEDRA codes, and frankly, some events just don't work in claims. So I think for all the reasons that have been brought up earlier, having a comparison group built on built on claims and or EMR data can be very, very problematic. Seriousness is a major one. We can't actually pull out seriousness definition from a claims database. So what does that mean if we needed to make a comparison there? Would we have to take all events from our clinical trials that meet that definition regardless of seriousness? So that's something. And the last piece would be adjustment. Understanding that the differences between a population we might derive from a large claims or EMR database that might reflect something that's closer to a general population may not really reflect that of our clinical trials. So bringing in at least age and gender adjustment, but other things as well. So folks, thoughts for consideration. And as I said, I think those claims and EMR considerations are important to think about, not only in terms of the clinical trials, but also in terms of how we would provide context for any ongoing observational studies.
Great, thanks. Salim?
I was brought up with the principle that when you can do things in a simple way, why complicate it? Now, categories A and B are simple. Don't complicate it. You know, whatever you think, just report it. That's rare, the way Janet put it. But when you come to category 3, it seems that the majority of people from industry are asking that question a different way. When you can do things simply, when you do things in a complicated manner, why even try to simplify it? That's the way all of you in industry are approaching it. Sorry, that's the way I feel about it. You can never interpret, almost never interpret category C information without context, a comparator, without context, which is the totality of the evidence. The effects on other adverse events, maybe it's going exactly the opposite way, and benefit. So I think the new version of this guidance should say category C does not have to be reported to us unless a comparator analysis by a group that is looking at the totality of the evidence feels it meets a threshold. But threshold, what does that mean? Threshold is not just a matter of absolute or relative value. It depends on which event. I mean, if I find a 5-fold increase in headaches, well, big deal. Even moderately severe headache, big deal. I'm not sure the FDA wants to know about it. I'm not sure it's going to change the way a program is going to develop. But if you take something more serious, then it matters. So it has to be on serious events. It can't be on other events. Once you do that, the safety reporting starts to become simple. And I would say on clinically important events, a excess that is clinically important. Now, how do you define excess that is clinically important? I'd say something of sufficient magnitude and seriousness that would change the course of direction of your development program, could seriously modify a trial, or in the case of an individual patient, would be seen as life-threatening. So you need something that is fairly defined, restrictive, but wherever possible, use your group data, comparator, not non-comparator data, and report it. And it has to be, and the DSMB should report it, when the net effects on health is adverse. So there's a judgment. So, for instance, in trials of thrombolytic therapy, if they panicked and reported every intracranial bleeds, we would never know it saved lives. We'd never know it saves lives. And intracranial bleeds is pretty serious stuff. You know, it is. But its rates is 1/20th that of mortality, and the net effect on mortality was highly favorable and became one of the most commonly used treatments for a long time, and still is in the majority of the world. So I think we need to step back, leave A and B as it is. C probably needs a substantial rethink. Now, the title of this symposium, which is staring at me, is Safety Assessment for Investigational New Drug Safety Reporting. So I don't want to bring into this post-marketing surveillance. That's a different issue. It has a different, I know it's useful in certain circumstances, but it's incredibly more complicated. So let's not even bring that into this. Let's just talk about, and usually this means there are randomized trials going on, and that's what we're reporting. But the word is reporting. Most of us in this room have focused the discussion reporting from a sponsor to the regulator. But there's another level of reporting from the investigator to a data management center or a coordinator, and then the sponsor reporting back to the investigator, and then to its ethics committee. That is a much bigger nightmare. If you think you're drowning in paper without context, Bob, you have much better context than an ethics committee has or an IRB has. They don't have a clue what's happening. The second thing is, this thing is drowning investigators. It is drowning ethics committees, and IRBs, and I think we should put a moratorium on it. Category C should never be reported to investigators or ethics committees unless this independent data monitoring committee says, look, it's crossed a threshold where the balance of harm exceeds the benefit. So when you look at waste in the entire system, actually 90% of the waste is the traffic between the center and the coordinating center back to the investigator, investigator back to the ethics committee, and then your auditors coming and saying, did you have enough oversight to make sure every one of them was reported to the ethics committee? If you could do that, that's a dramatic shift, and it would truly reduce the waste. And I know this last thing I'm talking about is covered by it, but wasn't on anybody's agenda. Nobody talked about it, but that is the bigger elephant in the room. So please, let's try to think about it. The last thing is, how would we, I strongly believe you don't need safety assessment committees for ongoing trials. Instead, you need the DSMBs or DSMB Plus, which is a program being monitored by a DSMB. Of course, that's going to raise new questions, which are good questions to deal with. Now, if you've got 5 studies going on, are you going to use the same efficacy and monitoring boundary as you would do with 1 study? You lose the chances of replication if you take the collective data and then you say, okay, I've now crossed 3 or 4 standard deviations or 5 standard deviations. You lose the chance of looking at subgroups, because the only way today we can look at subgroups is having 3 or 4 positive trials, and you pull the data from those positive trials. You minimize your chances of looking at efficacy versus safety and the balance of that in a more precise manner. And by having different designs, because no 2 trials in the same program are identical, you're actually doing more than replication. You're doing some degree of generalizability. So I like the idea of DSMB Plus, but let us work to come up with solutions for the new challenges that they will face, but they're good challenges to deal with. They're intellectually satisfying to deal with. So I'll stop here, and I'd say category C, stop getting it on an individual basis, collect it on your regular form centrally, send it to your DSMB. When it crosses a threshold where harm overwhelms benefits, then send it to the regulators. And the definition of that is it's crossed a threshold where you're going to modify your program. That's the threshold you would use.
Great. Gerald?
Okay, well, thanks. So I'm Gerald Alpan. I'm the head of the Office of Surveillance and Epidemiology at CDER at FDA. And we don't work in the pre-market IND space under these types of conditions. We're not looking at aggregate IND data. So I was a little surprised when I was invited here, and I had a little panic call to our internal organizer and said, well, you know, I don't do this stuff. I don't know what the threshold is. But let me give you some perspectives I have from the post-market. We deal with a wide variety of data. We deal with a lot of adverse event reports in our FAERS database, and the easiest, when we're able to use them, and we use them a lot for safety label changes, is when they're like type A things. And essentially that means the background rate is 0, or so close to 0. When we have the type C events, the bleeding with anticoagulants, et cetera, there's really not much we can do with individual reports. And we go to things like Sentinel, we go back to clinical trial data, et cetera. In the type B data, where these things aren't generally drug associated, but do have some frequency in the population, what's challenging, and I think what brings up challenges for this discussion, is how hard it is to really understand what the population frequency is of a certain adverse event in patients with a given disease. So we've had an advisory committee several years ago and about, I believe, some anti-diabetic products and the incidence of certain cancers in those products. And one of our epidemiologists did a great job of looking into this, but there's not some great library out there of pancreatitis and breast cancer and colon cancer and inflammatory arthritis that occurs in patients with disease A, B, and C. So it becomes very difficult to generate these kinds of backgrounds from the available data we have. They appear in claims data when somebody complains about them and when somebody bills about them. And that doesn't mean it's impossible and you shouldn't try, but there's no single library out there to get the right comparator data. Another issue is the time course of the adverse events. I saw that Ann's graph was in person years, and that's often a very good way to do things, but some adverse events are duration dependent. So you may see your first clustering in patients who've been on the drug a long time. If your person time is dominated by people who've just entered the trial, you may be misled a bit there as well. I think good planning is important, and the idea of having the blinded committee and the unblinded committee, having plans is important. And I assume that these plans depend on the drug, its known risks, its class, and what you're treating it for, because in the end, as I think some of our other presenters have said, this really is about balancing the benefits and risks of the drug, not simply determining absolute risks. What the comparator is important, as I think, as a lot of disease areas become very mature and the therapies that are available for them, I think it's going to be more and more difficult to get placebo-controlled trials, and a lot of therapies will be add-on therapies, so you're going to be comparing your drug to your drug plus some other drug and teasing out what that other drug is, especially if it has maybe a similar effect or a similar adverse event profile, will be difficult. The next question is, what is it you're going to do with the data that you have? And I think Mary Ross said, no one's interested in pulling all these, putting all these INDs on hold. That's not the purpose of this. Might you modify your protocol? Might you add some new testing? I think you have to think carefully about that. If you come up with really something unexpected, you might add something to the protocol to look more systematically at something that arose spontaneously within the context of the study. I think for drugs for chronic diseases, even if they have a placebo-controlled or active comparator-controlled trial, many of them often have open-label extensions, and if there are duration-dependent adverse events, maybe some different techniques might be used there. That's where the historical comparators might be useful, but also very difficult. I'm not sufficiently knowledgeable about SACs and DSMBs to make a comment on that. And if anybody was looking for the magic threshold number from FDA, I don't have it. Bob, do you have it? So those are my comments.
Yeah. Great. Thanks to all of the panelists for those comments. So one thing that I didn't hear much of, but I did want to ask the question around, but Anand, you touched on this a little bit when you talked about sort of the issue of multiplicity and particularly in looking at unanticipated SAEs. One question is, how do you sort of avoid sort of over-reporting unnecessary things? So when there might be an imbalance, but it's not meaningful and it should sort of be not reported versus the ones that really should be reported. Just views from not just you, Anand, but anybody on the panel on sort of how to grapple with that.
Well, I gave you a simple answer. Don't report it at all.
Yeah, well.
Let the DSMB look at it. I think you're asking the wrong question, Greg. That is the problem. And you will inevitably get the wrong answer. I'm not trying to be difficult. I think we've started off with the wrong question. The question is, when can you generate reliable information that is meaningful? If in the end you get data, and as somebody in the previous panel said, oh, we just tell the sponsor, you tell us what you're going to do about it. That's useless. It's like on rounds, I tell my residents, if you're going to order a diagnostic test, that diagnostic test better make a difference to what you're going to do to your patient. So in a sense, this is a diagnostic test. So if you don't have a plan what you're going to do, once you get the result, why even do it? So I'm very serious. I'm not being facetious here. And I think it would be a total waste of opportunity, this meeting, if we try to drill down on detailed nuances on the wrong question. It may be better, it may not have been part of the original agenda. It may be more productive to the scientific community and to FDA to refocus the question.
Okay. Thoughts?
I think it's difficult to interpret some of this data, but if you have processes in place at your company, so for instance, if you have physicians reviewing your individual cases, your clinical trials, and they see something, or something on it with another class is reported, or there's some literature article, and the experts in the company, which is generally the safety review team, look at that data and decide whether this is something that appears to be due to drug or not. It's a clinical judgment call. You can never say for certain whether it is. It's looking at everything that you have available. And I personally think that's the approach. And I do think that we have quite a high bar. For instance, at Merck, I cannot go and say that something is a serious event is something due to drug without having appropriate evidence to support it, because my senior management will go back and think about it again. So, it's science, it's process, and it's planning. That would be my answer.
We look at a lot of things all the time. I think to what Anne's saying, we have events that are being examined, every day, even if it's not in the context of a specific test. So I think the issue of multiple testing, I think, becomes particularly relevant when you're thinking about conducting a statistical test. But many times we're talking about simple percentage differences or rate differences. And so an absolute difference, I think we would need to think about all the different times that we would look at it.
And sorry, one other thing. I think it's important that, if we're going to send an aggregate IND report to FDA, we actually have to present it to our senior safety review committee before we send it down, and we have to be relatively convinced that it's due to drug, and we're doing something about it. I think that's how we prevent.
And that gets back to Salim. What you were saying is only report if it's significant enough to actually change what you're doing in the program.
And then if you work backwards from that, if we agree that is a guiding principle and then start to work backwards, then we say, okay, what kinds of things? How would we assess it? It'll then go back again to the comparative assessment. Because, you don't want to make major changes to any program without having some pretty good persuasive evidence to do that. So if you take that view, really 90% of this problem will go away.
And I think that view would argue against a single number in all cases, but I think it would really depend a lot on benefit risk and what it is you're treating, and our whole benefit risk framework, would come into play here, really making these kinds of decisions.
Okay. See, I told you. I didn't even open it up to the audience, and we're already getting a line. So this is a very participatory audience that we have. Thank you very much. Go ahead.
From Abby, can you hear me?
Sure, yes.
I want to comment on the assigned kind of topic that is the threshold, even though there might be a bigger question, but I want to comment on the threshold.
Yeah.
We have tried to implement 2 pilots in our company, and for that we have tried to establish 2 kind of thresholds. One is statistical, the other epidemiological. The epidemiological, perhaps, as you have pointed out, may be based on electronic medical records and all, maybe not that precise because not necessarily resemble the database, the clinical trial data. But at least we can use it as a parameter to measure against available other data we have from other clinical trials, perhaps in the same therapeutic area. Then it helps you to at least identify around what number is the threshold. The other is a statistical kind of threshold that maybe it's not 10 versus 1. Perhaps it's a little bit more complex than that. But we use both of them to try to establish a threshold to see if an event need to be reported or not. A bigger question again is, shall we do it for all clinical trials and for all the products? That's something that a little bit concerns me because of the amount of resources we take into this activity. Just generating the threshold, it took about months to get all these. Then to do it across every single therapeutic area is quite difficult. Assistance is provided enough by the FDA, but perhaps by other organizations about some thresholds, at least for major conditions, that would be great. Thank you.
Any comments from the, or in agreement?
I don't see the point of trying to define the threshold because, quite honestly, whatever the threshold, you got huge uncertainty.
Yeah. Peter?
Well, I'm also not going to try to define the threshold, so start with that point. I just wanted to put a perspective forward, perhaps as a counterpoint to a certain extent. The intent with this is as a starting point to avoid the excessive reporting that we were seeing before, where everything that was listed as drug-related was sent in, regardless of whether there was any rationale to think it really was. And that kind of over-reporting of events leads to, unfortunately, really a signal-to-noise issue. How are you going to figure out which of those hundreds of events that get reported actually are meaningful so we can do something about those events? When we're thinking about the type A and B types of events, though, I think we've all agreed that's not terribly complicated. B, maybe sometimes a little complicated. It's clearly the type C type of events that we're trying to understand. Well, why are we trying to understand that? We're not talking about routine, non-serious adverse events. We're not talking about tolerability issues. We're really focused here on serious, meaningful events within a trial where we believe there could be a causality. The drug is leading to an imbalance, an increased risk that's important, that's clinically relevant. Why is that important? I mean, it's important for pretty evident reasons, but let me just make sure that I put those forward, because I don't think this is a risk-benefit issue per se, although it comes back to that. So I'm going to try to connect that back in just a moment. I guess the perspective I'd put forward, and be interested in the comments, is that what we're trying to do here actually is to protect the trial and to protect the patients. We want to make sure if we have a serious risk that was unknown, that's induced by the drug, that patients are aware of it, and particularly important that investigators are aware of it, because they can potentially mitigate, monitor, assure the continuing safety of the patients in the trial. I think we heard earlier about I think Ajay mentioned the GSK drug, where there was a question balance in renal insufficiency events. I think that's a pretty good example. If you have a drug where that's not known, and during the conduct of the trial, it becomes evident that there really is a signal. I'm not talking about 5 versus 0, because that may or may not be real, but where a careful, thoughtful analysis, what you were mentioning, where an appropriate group looks at the totality of information, perhaps from non-serious events, from the laboratory data, but from the serious events and their careful scrutiny, and concludes that the drug has an effect to induce events of renal insufficiency in patients who are at risk because of dehydration. I think investigators would need to know that so they can properly manage their patients, monitor their patients, and help to avoid and mitigate that risk. That potentially can maintain the trial, keep its integrity ongoing, and actually see whether there's ultimately benefit that outweighs that risk. That's what I think we're here to try to figure out, which is how, in the context of ongoing trials, where there is a new safety issue, and I might point out, I think this is generally going to be, type C events, I suspect, are going to be the least frequent. I think very commonly we know the profile of drugs before we get to a phase 3 trial, and those are expected events. They're in the IB, they're already in the consent. If it's second or third in class, it may be because we know it from prior drugs within the class. So very frequently, these are not going to occur, 20 of these type of reports, I certainly hope, in the context of a trial. These are infrequent occurrences, but we're talking here about serious, important events for patients and physicians who are investigators to be aware of. And again, I think to keep the integrity of the trial going, we want to be able to make sure that patients are properly monitored and managed in the context of the trial. So just wanted to clarify why we're doing what we're doing. What we're trying to figure out is, and I think we heard some very thoughtful comments, when do we think that's a real event versus when do we think it's the 5 versus 0 that's just noise and doesn't need to be reported? That's not so straightforward, of course. But thanks, and be happy to hear any comments from
Yeah,
from
great framing. Any comments on that last? When is it a real event?
I don't know about when it's a real event, but I think that I would like to add that, to find those relatively few, we kind of need to look at everything. So it's just that's just the flip side of it. The specificity is going to be pretty low.
You never know whether it's a real event on an individual case. But even on an individual case, you can put some threshold so that you're not drowning in noise. And it could be, you can have a certain level of seriousness, you can have a certain level of causality, but one of the most important thing is the physician has stopped the study medication. If they continue, it means in their mind, whatever they say, it's not linked. That again will substantially lower the numbers that you're going to report. So once you put it all together in a well-run trial, at least in cardiovascular disease, depending on the duration, people may stop drugs at about 10% per year. Half to 2/3 is because they just don't want to take the medicines. But about a third will be they think there is some side effect. So you add a level of seriousness to it, and the sponsor collects it, but you don't report that to the regulator. You then report that to your DSMB, who will then take that data and will make an assessment. That will then help you. So at every level, think of streamlining it. Thus far, the approach is, I'd rather know anything that's happening because I don't want to miss anything. The problem with that is when you're drowning in noise, you can't see the signal.
But, I think FDA, with the individual case reports, individual SUSARs, you should only be sending down to FDA if you, as a company, think there's a causal relationship with that specific case. So, for instance, at Merck, with our IND reporting for serious adverse events, we don't send most of the cases down. We only send if we think it's due to drug. And that would obviously then we look at it from aggregate point of view so that you have, this is part of the next step. So I think that, apparently that has helped a lot, especially with the oncology studies, so they're not completely overwhelmed with all those SUSARs. So we have implemented that.
Great. Next question.
Yeah, Janet Wittes. So it's very rare that Salim and I disagree, but in this case we do. It seems to me that if one follows what the guidance is saying, you'll reduce the paperwork by 90% automatically because of what Anne just said. There has to be some causality. There has to be the difference between the treated and the control group has to be big enough for it to be convincingly bad. So I think the guidance actually, if people followed the guidance, then there would be a lot of decrease in reporting. In our paper that several of you cited, and I feel like those of you who are statisticians are going to say, why in the world did she do this? We said, forget multiplicity, because you can't deal with multiplicity at all. There's too much. Think of it on these individual bases, individual events, individual events that are merged a little bit, not preferred terms, but high-level terms so that they're things that are like each other are put together. And that's the basis for making a decision about reporting. But it's not, the idea is not to stop the trial. The idea is not necessarily to change anything. It's to provide the regulators with some information, because remember, they have information from drugs from the same class to be able to put things together in a way that nobody else can. So the other thing that I wanted to say at the last session is about DSMBs. And I try to say this in my presentation, that DSMBs think of risk versus benefit. And this is not a risk versus benefit question. This is a risk question. What is the drug? What are the adverse events that the drug is likely to be causing. And so if this becomes a responsibility of the DSMB, it will require a major change in their culture, in their way of thinking about, because the question will be, what is the risk independent of the benefit, and that we should be reporting out. Right now, I think what DSMBs do is be very cautious about reporting something out because they don't want to hurt the study, and if the balance of benefit and risk is good, they'll be quiet. Only when something is so bad that you have to change the informed consent, or you have to change what the docs know, and only when they're really pretty sure, and you like my technical word, pretty sure, that what you're seeing is real, only then do they report out. If they are to take the responsibility as the guidance describes, they will have to be reporting in a very different way, and I think that's a very large cultural shift.
Great. Any comments on that, or?
I'd say let's have that culture shift.
Next question.
Robert Baker, Lilly. First, before my question, a clarification to the point that Dr. Wittes just raised. At least for our company, we mail to investigators and IRBs based on the controlling regulatory body. So indeed, the FDA's IND reporting rule is making things better for IRBs and investigators in the United States, whereas the flood persists in other geographies. My question is actually a repeat of the one I asked the first panel, but I think would be quite relevant, probably echo some of what Dr. Youssef has been saying today, which is it'd be useful as we get to final guidance to step one step higher to really articulate clearly what we are trying to achieve and what we're not trying to achieve. This is really for the agency, I guess. And what Dr. Stein just said, I thought was quite clear and aligned with how I would think about it. It's not necessarily clear to us in the draft guidance, nor although perhaps I misunderstood it, Dr. Temple, I'm not sure it was exactly what I took from what you said before lunch, because I think for us the question is this about the subject protection, or is it a bigger requirement in our drug safety evaluation? So in CIOM's sort of terms, well, firstly, for subject protection, it's a point in time. Eventually the studies end, and for product level conclusions, we will have better data, we'll have better analyses, and we'll have free unblinding of all the experts to make those determinations. So if we approach this instead about protecting subjects in the study, then in CIOM sort of terms, we would approach it of looking at imbalances, but not trying to find and report all imbalances, but trying to find and report important potential risks would allow us to immediately filter events that aren't important and then have a secondary judgment that the data, as we look at this, add up to a potential risk and not simply a numeric imbalance.
Thank you. Next.
Listening to some of the reactions, I think there's at least some people that are adhering to an efficacy mindset here. We need to reimagine this with a safety mindset. Efficacy, it's testing and confirming with p-values, and p-values don't really make any sense here. If we did 100 tests blind, we're going to be wrong 5% of the time. You can make adjustments with FDR or something like that. You're going to be wrong some finite amount of the time. What we want to do is we want to leverage the multidisciplinary nature of the safety management team. Use statistical experts, use clinical experts, partner together in really trying to understand these safety issues. So if you recall the plot that was up there, for example, you'll notice that it didn't have just one threshold, because if it had one threshold, that would imply that you cross that, then you have to do something. So that's like a decision rule. We have 3 thresholds on top, plus 2 reference lines on the bottom there. So you have the background rate, you have some critical rate determined by the multidisciplinary team, and you have 3 thresholds there. And at any point in time, you can see where you are, the level of evidence for a risk elevation at any point in time. And across time, you can see the trend. So we're not trying to come up with a test. We're trying to come up with a quantitative framework to help the whole multidisciplinary safety management team understand the evidence and the data and drive the decision-making process with medical judgment. So we can't take efficacy analyses and realign them for a safety analysis. We have to develop them from the ground up here.
Great.
Sorry, not a question again.
It's okay. We don't have time for answers anyway, so Bob.
This isn't anything that Janet and Peter haven't already said to some extent, but I want to be explicit because Celine keeps bringing it up. One of the major purposes of rewriting the rule was to do exactly what you wanted to do. We were getting all kinds of crap because people would just send it in. I gather they still do that for the Europeans, and I guess they're happy enough with it and don't care. We wanted these large numbers of things to be screened for the likelihood or possibility, reasonable probability, whatever it is, that they actually were something the drug did. So the whole point of it is to do exactly what you want everybody to do and not report it. Not only not report it to us, but when it gets reported to us, it has to then be reported to all investigators who then report it under their obligations to report it to the IRBs, who then put it in the garage and never looked at it. You know, we knew that. But the primary purpose was to do exactly what you wanted to do. I just want to be sure everybody knows that.
So we're on the same wavelength. Can I?
Well, we are out of time for this session, so like 30 seconds to you and 30 seconds
okay,
to
the one
Chris.
thing that this DMC plus doesn't quite solve is the idea that across different compounds in the same class, how do you look at it? This could be an argument for getting things to the FDA sooner than later. The only problem with that is different agents of the same class, especially on adverse events, can have quite different profiles. If you remember Zymelangatran and liver failure, it wasn't seen with the other agents. So I think there you want to look at it class by class, because with Zymelangatran, had they taken all their trials and looked at it, they would have seen the hepatic failure thing. So I'm not sure that that level of looking at an entire class across different sponsors is that important as you're developing a program. Sure, at the end of it, you would. So I don't think that should wag the tail of the dog here. Okay, great. Then we're in agreement.
Okay, good.
Let's go home.
30 seconds to Chris.
So just a
and then
brief
that'll be
comment to
last
have it
comment.
on the record for international harmonization. And Bob, you mentioned this, but this is a reason we get from some sponsors on global trials for the need to collect SAEs in an expedited fashion is like B Farm demands it, and they're a company that needs the German market, and they don't want to upset them. And so we really need also to bring in our international regulatory colleagues to help make sure we can implement these more rational guidelines. And I think we all want to give the FDA a lot of credit for being the leading regulatory authority in the world for thinking through how to do this more rationally.
Okay, great. I'd like to thank the panelists and the presentation. We're going to go ahead and take a 10-minute break. If we want to finish on time today, be back here exactly at 2:45 for us to start. Thank you. Okay, welcome back, everybody. In this last and final session, we're going to discuss key data and methods issues associated with conducting aggregate analyses of patient-level data pooled across the study arms and even multiple studies within a program, as we've been talking about all day long. Joined with me on this panel are Mary Nelson, Research Advisor, Safety Analytics, Global Statistical Sciences at Eli Lilly and Company, Asif Haq, Director of Patient Safety at AstraZeneca, Bill Wang, Executive Director, Clinical Statistics at Merck Research Labs, and Mark Levinson, Director of the Division of Biometrics 7 at FDA. I'm going to first turn it over to Mary, who's going to open up the session with a presentation, followed by panel discussion, and then we'll turn it into the audience discussion as well. Thank you. Mary?
I'm happy to be here to talk about data pooling. I'm actually quite excited that this ended up as a session at this workshop. I think sometimes this topic gets underappreciated, so it's nice that we have an opportunity to talk about this. So I'm going to talk about some of the common pitfalls that we sometimes see when pooling data from multiple studies and talk about best practices and trying to avoid these pitfalls. And I'll also talk about how Lilly incorporates best practices in the IND safety reporting process to avoid these pitfalls. So as we've been talking about, we've all been focusing on type C. This presentation will also focus on the type C type event. So we're talking about the aggregate analyses. I'll be focusing on the case where you have a concurrent control, and also the assumption is we will have unblinded data. So I guess the challenge we've been dealing with through this whole workshop is trying to figure out how to right-size this effort. As already been mentioned, we know that it's a difficult thing to try to figure out whether an event is reasonably likely caused by the drug. It's very difficult even when you have all the data at the time of the submission. So when sponsors put together a proposed label, even when you have a lot of data, it's very difficult. So you get aggregate analyses from statistical tables, but it also involves maybe multiple aggregate analyses. Or it includes a lot of medical reviews. You look at events that are similar to each other. Or you look to see if there are events in drugs from the same class. You look at all sorts of things when trying to figure out if an event is reasonably likely caused by the drug. So how do you right-size that effort when you don't have as much data? So the point of this presentation isn't so much if you do that during the IND safety reporting process or who does it, whether it's an internal group or a DMC or a DMC-like group. The main point of this presentation is trying to say whoever conducts these aggregate analyses, it needs to be done in a very thoughtful manner. We don't want to get in a situation where decisions are being made using summary tables that fall victim to some of these very common pitfalls. So one pitfall when someone tries to simplify this process or try to make it easy to look at data from multiple studies is it's very tempting to just take all the studies in a clinical program and just pool it all together. What happens in this case, you can get in a situation where study and treatment or treatment arm are confounded. You may end up with a table where one treatment arm comes from one set of studies, another treatment arm comes from another set of studies, and when you have that, basically study and treatment are now confounded. So when you see a difference, you don't know if that's because of treatment or it could be because of study. More importantly, if you don't see a difference, that treatment difference could be washed out because of a study effect. So here's an example of one pitfall. This example comes from the literature. It comes from a 2002 article, so these pitfalls have certainly been around in the literature for a while. The problem we're seeing is not everybody is aware of these pitfalls, or the right people who are conducting some of these analyses may not be aware. So in this example, there are 6 studies. 4 are placebo-controlled studies, and 2 are active controlled studies. So if someone tries to simplify the process so much and just simply pool all 6 of these studies, you often get a table of events or look at the last row. You might just look at a total count and a total percentage. So in this case, you might see the percentage of this event is 1.3% for your new drug and 1.3% for a placebo. You'll look at that and say, great, that's even. I'm going to move on to other events. But unfortunately, the underlying data will tell you a different story. If you just looked at the 4 placebo-controlled studies, you'll see that at least 3 out of the 4 studies actually showed an increased risk. So by pooling this data, you lost that signal. The next 2 examples are just trying to show that even when dose arms are confounded with study, you can get into some misleading results. In this case, the placebo group, the estimate comes from 2 studies. The low dose group comes from 1. Middle dose comes from 2 studies, and the high dose group comes from 1 study. Within study, it's very clear that there is absolutely no dose response. They're just equal across doses. However, when you pool this data, it actually looks like there is a little bit of a dose response. And so if all you're doing is looking at this last row, you're going to have people trying to look into this event further and basically probably wasting some time trying to look into this when really it was just an artifact of how you pool the data. This is very similar, just dose arms are confounded with study, but this shows that it can go in either direction. So this is an example where the first study does show a dose relationship, the second study shows a dose relationship, but when you pool the studies, it actually gets a little less clear. That it just kind of jumps around a little bit, and it may not catch your attention quite as easily. So another thing that can happen when you try to simplify the process too much is you can create a summary in such a way that you don't account for study differences or you don't adjust for study. So even if all your studies have the same arms of interest, you can still run into a potential pitfall. So here's an example of that. So here are 3 studies where you have, all 3 studies have your drug and a placebo control. So you think you're great. Well, you're directly comparing these things, and sure enough, you can run into yet another problem. You avoided the confounding problem. You're not confounded with study. However, you still have an issue because of unequal allocation or randomization across these studies. So even though the percentages are the same in each one of these studies, when they're pooled, it actually looks like the percentages are different. And this is the Simpson's paradox that has been brought up already in this workshop, is mentioned as a potential problem to look out for. So what are the best practices? So again, it doesn't really matter who's doing this or when it's being done. These are best practices that should always be thought of when pooling data from multiple studies. We should just try to avoid confounding when we can, and we usually can. I think if you don't have very many placebo-controlled studies or active controlled studies, you might be in a different situation. But for most compounds, you can avoid confounding. You also need to adjust for study in some manner. Now, this could be done various ways. One way is to just never pool studies and always look at just the individual studies. You can just always look at that. Now, that's not a bad way to go about it if you're just looking at a handful of events or you know which events you're trying to follow. But if you are trying to create a system to monitor a whole bunch of events, like if you are trying to look at all SAEs to see if there's anything within a balance, that might be difficult to do when you have a lot of SAEs to look at. A forest plot's very nice. It's another way to look at all different studies and then maybe a pooled summary. However, that is also can be challenging if you are trying to look at a lot of events, but it's a very reasonable approach if you don't have very many events you're looking at. Other techniques include study size adjusted percentage, Mantel-Hansel odds ratio, Mantel-Hansel risk ratio. These are all methods that can be used to help adjust for study. Now, I know p-values are controversial or confidence intervals are controversial, but some of these can be useful to at least help you determine if you're in a paradox situation. If you have a Mantel-Hansel odds ratio of 1, but your percentages look different, that will tell you you're in a paradox situation, and the other direction can occur as well. So most of these methods for adjusting for study is more in the context when you have a fair number of events. If you are talking about just a handful of events, or if all your events are occurring in an uncontrolled phase or extension phases, then different things are needed. So you may need to just look at the individual cases and medically review the cases. You might be able to create an exposure-adjusted incidence rate for the entire exposure for your new drug, and compare that with literature and do the best you can, or some background rate. So that gets a little trickier, but at least when you have a fair number of events, we can certainly do better than some of this crude pooling. So let me just take one of the examples I mentioned, the study size adjusted percentage. That's one technique that can be used to help avoid some of these paradoxes. In this case, we are going back to this situation where we have 3 studies, all have the new drug and placebo, and all the percentages are the same. So if I crudely pool this data, it looks like the percentages are different. However, the study size adjusted percentage actually just weights the data in a slightly different manner. It makes sure that the weight of the new drug and placebo get the same weight within a study. It still allows for the larger studies to have larger weight, but at least within a study, it ensures that the weight provided to new drug and placebo are the same. So when you use this technique, you'll actually see the percentages end up the same. It's 26% and 26%. We think this particular method is probably underutilized. I don't think very many companies use the study size adjusted percentage very often, but it is a pretty intuitive method to try to get more and more into predominant practice. So briefly, how Lilly incorporates this thinking into our IND safety reporting process. In our case, we do have surveillance teams and safety monitoring teams already looking at blinded data. And when there is an event that they need to have unblinded, or if it matters that it needs to be unblinded, then it goes to a safety internal review committee. So this committee just receives a small number of events. They're not trying to look at a whole bunch of events at one time. In this case, the data that's given to this committee comes from the study team, the cases and the exposure numbers. The denominators by treatment and by study comes from the study team. And for the blinded studies, the treatment arm denominators are then estimated. We're not unblinding the studies just to get an exact denominator. And then a different team, the case management team, can give the treatment assignments for the cases to this committee, and then this committee will then run the analyses and determine if it's a reportable event. In this case, it's easy to help keep these best practices or avoiding these pitfalls because it's a pretty small committee. We have very experienced statisticians on the committee, and they know how to look at the data. They know to look at the data by study or to create some of these more advanced methods if needed. So just in conclusion, no matter who runs these analyses or how often or whenever they are done, the idea is that whenever they are done, it should be done in a thoughtful manner, and statistical thinking is still very important. Unfortunately, we still have a pretty big educational gap on these pitfalls. Maybe people here are aware of these, but I could tell you a lot of people who are conducting these analyses are not aware of these pitfalls. And I'd say the educational gap is cross-functional. You might think all statisticians should know this, and that may or may not be the case. I certainly did not learn this in school, or if I did learn it in school, it didn't sink in. Now, of course, that was a while ago, and maybe it's getting better, but we still see statisticians coming into industry without the knowledge of this kind of thinking. Sometimes it is on-the-job training when it comes to these kind of lessons learned. Certainly for me, I was really early in my career. One of the first things I had to do is look at an adverse event across multiple studies and created a table. A colleague of mine and I went to my senior director and showed the data and said, hey, this looks great. It looks like our event is the same or better compared to placebo and active control. And the first thing he said was, are you sure there's not a Simpson's paradox going on? And I'm like, what is that? So I had no idea. And his name was Charlie Sampson, so I thought he said Samson paradox, right? So I just kind of giggled, and he's like, why are you giggling? I had no idea what he was talking about. So I learned pretty quick what a Simpson's paradox was. But not all statisticians will. A lot of statisticians are in the world of individual studies for quite some time before they get into the chance to look at integrated analyses. So it's not something that you should assume your statisticians know. So we do have an educational gap. So for the people who do know these things, I guess maybe help with the education. the idea is whoever's doing these analyses, we need to be very thoughtful. And there is a need for more innovation to try to make some of these methods easier to do. And that's true not only for the IND safety reporting process, but even at the end, at the submission time. We need to do better about making this easier to get these better methods used more broadly.
Okay, thanks, Mary. We're going to go ahead and turn it over to Asif for some comments.
Yeah, thank you. A very lovely presentation. I think your presentation really highlights the pitfalls and the challenges that exist with pooling data from different types of studies, some of which are placebo-controlled, some which have an active arm, and also duration of exposure studies. Without unblinding cases in the safety database for aggregate analysis, we can look at also patient exposure and calculate the incidence rates and also look at literature rates, which you already highlighted. While I'm not a statistician, but I still read up a little bit and learned real fast, like you said, about the best method. So I came across a really interesting article, a literature article by Jackson et al. In Wiley Statistics. And this talked about the best measures to meta-analysis in trials of differing types. And one of the measures which they have suggested is that the random effects meta-analysis, which can be used as the random effects model, because this model will allow for true effect variation between study and study, and also within the study, and also attempt to correct for it. The odds ratio and relative risk will be similar, but if the event rate is small, therefore it will not give a correct answer. The pooled estimate of the log odds ratio and the corresponding confidence interval can then be transformed into an odds ratio scale. However, the approximation made by the conventional random effects model can be poor when the studies are small or the effects are rare. The random effects approach also allows generalization to the wider population, and the fixed effects approach does not, which is a significant limitation of this approach. I think we need to decide upon, hopefully with the agency's approval, of a consistent approach to how to match and how to look across studies which are blinded and unblinded, and also for differing types. That would be my thoughts.
Okay. Great. Bill?
Yes, so first, thank you and congratulations, Mary, for point important issues. In some way, I feel this pooling is like great quantitative tools. And it's a tool like a hammers. And sometimes when you have a hammers, we tend to find the nails. I think Mary's talk really gave a great example of that. If you don't use the camera carefully, you may hurt yourself. You may not only not hit the mirror, you actually can hurt yourselves. So that really makes me try to link to what we are doing in this effort in the safety assessment, and link to some of the work we are doing at Merck that Anne presented, but also link to some of the things we are doing. Quite a few people from our NSA safety working group in the audience. I'm happy to be a co-chair of that. Some of the work we are doing in thinking about different things. I think there are a couple of things when we think about poolings. Once we have to understand what question we try to solve. It's important to really clarify the question, to get the question right, to really clarify what is the nail. Here we have a lot of talk on whether it's generally using the wheat versus chaff. Some people are using the chipmunk versus wolf. Some people I sometimes use my paper. I have a paper talk about canary in a coal mine. But some people are using benefit versus risk. And I think, Bob, your first talk is about whether there are evidence to suggest there's some relationship. And I think Tom used the ones, the signal versus versus the noise. At the end of the day, I think it's really need to clarify, even though in the last session I feel we reached agreement, but I think there's still some ambiguity there. What exactly is the question? Is it a risk question or it's a benefit risk question? And that actually is important differentiation we need to clarify. I think, Janet, in your comment from the audience, you think it's a risk question. And some people think it's risk-benefit question. I think it's very important to clarify that question. very critically important, because that will determine what you're going to do, how you're going to pool. So that's why in Anne's presentation, this first box of accept process is to clarify the questions, to clarify what are the population, to clarify what are the variable of interest, to clarify what are the contexts that question is, the intercurrent event or the background, and to clarify what's the metrics. That's the important of the first box. We call the safety estimate. And we need to clarify that in order to get a good evaluation, because when you have that question clarified, you can actually plan for your pooling. You can plan what population are we talking about. Are they pollable or not? What event are you polling? Are they adjudicated so you can actually pull them together? And then what are the intercurrent background they actually make it pollable? So that's an important part, to ask the right question, to plan for the polling. Then when you do the pollings, there's actually many ways of pollings. We need to be careful and we need to really think about. There's polling across study. Mary talked about that. There's polling across time dimensions. I think there's a few examples say polling early versus polling late actually can make a big difference. There's also a question about polling across subpopulation. And I personally work on ICH E17. We spend a lot of time talk about polling adverse event across regions. And that early planning is very important in order for you to pull the event together. And of course, in this particular setting, it's really ongoing of the pollings. So there's a couple of factors we have to also consider. There's that speed of data collection, the so-called the velocity. There's also the volume of the data collection. If you look too early, there may be a risk. But if you look too late, it defeats the purpose. There's also the variety, different type of adverse event. Then also the quality that Dave, I think Dr. Metz mentioned very highly. That's something we really need to pay attention to make sure that data are collected and cleaned in a timely manner and coded in a timely manner. So when you pull, it's pulled in a meaningful way. Because sometimes you need to pull across, preferred term into a high-level term. If you don't code them, you cannot do that. So you cannot make those assessments early on. And I think another important part is when you try to make decisions using pollings, it's not just going to be one number help you make that decision. There are many ways you can do it. Single number is something we get used to, but with the technology, we can do a lot of things with poolings. We can do polling, we can do dynamic, we can drill down, we can roll up. All those things, I think, can be important part of this polling exercise. That's why we don't feel the quantitative framework is important. Have some kind of threshold guide you is important, but it's not going to be one number. It's a set of framework, allow you to look at the data and be able to look up and down, pull left and right, to make you that holistic judgment. And that's also why in our ANSN safety working group, we actually have multiple working streams working on all this kind of thing, including a dynamic visualization. I'll stop here.
Thanks for all of those comments. I really appreciate it. Mark?
Okay, well, thank you. First, I'd like to commend the industry statisticians, both on the panel and we heard in the audience. I think they're providing a lot of leadership in this area to flesh out the statistical aspects of this guidance, and I think we've heard a lot of good ideas. I think a lot of what I'm going to say has been said, but it probably functions as a good review here. So I'll point out mainly challenges and perhaps a few solutions. First, I'd like to point out that we do have an upcoming guidance on meta-analysis for safety. We've been working on this for some time. Lisa Lavange worked on it during her years at FDA. This guidance actually addresses a lot of, it's not focused on this general surveillance problem. It's focused on drug safety when you're trying to evaluate a small number of hypotheses. But a lot of the principles are, I think, overlap, and I'll review some of those principles, and they have been mentioned a lot today. First of all, one of the biggest principles is to stratify by trial, which was really the subject of Mary's talk. Crude pooling, especially in the circumstances we're into, can lead to all sorts of problems. And I think Mary demonstrated many of those problems. Another point we make in this guidance is on outcome ascertainment. We need good, consistent, valid estimates of outcome. The more homogeneous across trials, the better. But a lot can go wrong if you're not focusing on obtaining the outcome correctly, hopefully prospectively, but that's not always possible because sometimes safety events are sort of discovered retrospectively. What we heard, I think, from Bill just now is the importance of planning. Planning and pre-specification. Now, that's sometimes difficult in safety. It's certainly not the same sort of planning we might see in efficacy, but the more upfront thinking and pre-specification is going to go a long way to making the process more objective and hopefully improving the process. So pre-specification and what trials to include, what outcomes to look at, the analyses, the thresholds, all that is very important. Another point this guidance makes is on the totality of evidence. In safety, I think we've heard this a few times today, a single p-value is often not very useful. So the totality of evidence would include like the effect size, the consistency across trials, and also the plausibility of the results. So that's another point. And finally, last point I think that's relevant from the guidance is having the appropriate expertise. So that includes both the clinical, the epidemiological, and the statistical expertise, all in place, all to draw from. And another point, and I'm sorry, one more point, is the clear objectives, as Bill pointed out. Have clear objectives up front. Okay, let me move on to go beyond guidance, which hopefully will be available somewhat soon, to some of the challenges. And again, we heard a number of these challenges. And the first challenge is pooling across different study types. So this may be different patient populations that are different baseline risk. That presents challenges. Pulling trials of different designs, particularly different follow-up. Do you combine a short-term trial with a long-term trial? Well, it probably depends on the outcomes you're interested in. And it's important to use the right statistical measures that don't mess up the combination of short-term and long-term trials. For example, exposure-adjusted estimates, or maybe you're looking at hazard rates or so. So different follow-ups presents the problem. I think we heard something about extension trials today. Often, from our experience, we see safety issues, at least the counts appear in the extension trials. Perhaps that's what they're there for, this extension trials, is to get the long-term safety information. But they're often uncontrolled portions, and then we're often left with a bind that we never really get out of. Another issue with combining trials is what we call the small versus big trials. We often see in the kind of meta-analysis safety world, there's some big trial that highlighted some issue. So let's do a meta-analysis that combine this big trial with a whole bunch of small trials, and basically, because the big trial dominates, you're just getting the same result back. So it's important that the information is not diluted. That's, if small trials are relevant, they may need to be looked at separately. And of course, you may, at the end of the day, not choose to pool at all. That you may feel that the trials are so different, they have to stand on their own. But nevertheless, looking at things like forest plots and other summaries across trials is still useful. So then there's a challenge with ongoing trials. This came up throughout the day with terms like multiplicity and multiple outcomes. And I believe there was comments that we're not trying to do efficacy here. So the standards are different. But nevertheless, this is a challenge that we're ongoing trials. We're probably looking at them repeatedly, and we're looking at multiple outcomes. So we need systems and methods to address that. And we heard things like false discovery rate. There's things like cumulative meta-analysis that's designed to address this. And of course, sequential Bayes handles this well. But again, this is another area where pre-specification can help. If you decide ahead of time how this is going to be handled, then it becomes less arbitrary during the process. Brings me to the last challenge, which I don't have much to say about, is the challenge of using blinded data. It's almost impossible to aggregate blinded data by treatment arm, naturally. I mean, you can come up with like worst case scenarios, like if this fell this way, in this case, you can play around with bounds, what the worst it could be. The idea of aggregating with blind data isn't not a very reasonable thing to do. So there, you're probably going to be in that situation that was described with the two-stage approach. That you might look to see if their overall rate is beyond some threshold that would, force you or encourage you to unblind the data. So just two final comments. Again, we're not in a confirmatory exercise here. This was said earlier, but we can accept false positives. This is not the end of the story. And even if it's a false positive forever, it doesn't mean necessarily the death of the drug. So we're not in the same situation as efficacy. We're willing to accept some mistakes, and hopefully those will not have major implications. The alternative is a balance between, of course, the false negatives, is not finding important safety issues, so of course that's always a balance. So I'll end with something I've said throughout, and we heard on the panel, that upfront planning and documentation is important. What events that you're going to consider, the trials that are going to include it, the thresholds, the analyses, the procedure, when to unblind. The more thinking up front, the more objective this process will be. Thank you.
Great, thanks, Mark. Very comprehensive review, and looking forward to that guidance to come out. And I appreciate the sort of the range of challenges that can come in with pooling that you brought up, pooling trials of different patient populations, different study designs, different follow-ups, including ongoing and completed, all of those things, and even different trials of different sizes. But Mary, you pointed out a lot of the pitfalls in doing this, and the study size adjusted method might address that latter one. But let me get a sense from the panel on where are we in the state of the art of analytic tools for pooling in terms of really addressing these challenges. Mary, you did mention that there are tools available and there are really good ways to do this. It's just educating the statisticians, getting the best practices out there. But are all of the best practices there? Is there still room for improving on the analytics side? Do we need new tools and better tools to pool these data in a more appropriate way? Thoughts on that? You don't have a microphone. Don't you have, weren't you, oh, okay. Well. Okay, why don't you stand here? Okay, or go like that.
I do think there are a lot of tools available today that are underutilized. I think that's true. The study size adjusted percentage is something that we're not aware of too many people using that at all. I'm part of a FUSE working group, and it's a FDA industry academia collaboration. And there is a project team within those working groups working on standard analyses for safety data. And there's one white paper, the adverse event white paper, that does advocate for a broader use of the study size adjusted percentage, just in general, when doing aggregate analyses. But even more so, it could most likely even be used in labeling. Some of the percentages in labeling come from these crude percentages. So even in our labels, sometimes we have percentages that could be approved upon. So we think that's a pretty good technique that can be used today. The Mantel-Hansel methods are great today. I think the challenges that still could use a lot of innovation is when you don't have much controlled data, what you can do. I think that's what you need to do when you don't have controlled data, I think is definitely open for a lot of additional innovation and thought and how best to look at the data when you're in that situation. But for controlled data, I think we have a lot of good tools already available, and I think it is a matter of just trying to get some of these practices into predominant practice. And we're trying, this FUSE working group, we have the white paper. Also, my supervisor, Brenda Crow, created a YouTube video on what the study size adjusted percentage is. So it's a 10-minute video clip that's intended for a broad audience. It's for even non-statistical audience. So it's a pretty easy video to understand. So I think a lot of it has to do with education, but definitely more innovations needed, when you're outside of the controlled setting.
Right. Yeah, to follow on to what Mary just said, I think the need of the hour is consistency and agreement on what exactly is the approach that's required. Like you just said, there's so many approaches available, there are tools, but again, specifically when we come to the safety assessment for an IND safety requirement, at that time, we need to know exactly what we need to do and which would be the most acceptable for regulators. Because when we talk to our stats department, they'll come back to us and ask, what do you think is the best way to do? Because there isn't an aligned view right now. So I think the need of the hour is just to come to an agreement on what would be the best way to do it. Thanks.
So when think about tools, many times we get to think about the tool about uncertainties. But I actually feel a big opportunity there. It's not only about uncertainty, it's really clarify the ambiguities. So when I think about tools, I think about tools in a more general sense. Do we have tools to help us make things be more clear and clarify the questions first before you think about how to address the uncertainties? So for that, actually, I feel the recent work on the ICH, particularly on the E9 side, using the estimate framework, even though they focus on the efficacy, but that framework is very helpful even in safety settings to really clarify the question. In particular, in this setting, are we talking about risk or we talk risk versus benefit? So I think that type of tool, that general framework of tools is important to think about before we get down to the uncertainty tools. Now, on the uncertainty side, with all the technology, with all the visual tools, there's many things available, and particularly in the area of dynamically visualize things, to see things in a so-called dashboard way, and to be able to drill down, roll up. That, I believe, can really help a lot in the decision making. Not to mention a lot of statistical tools.
Great. Question?
It's a comment. This session is almost deja vu. I could have rewound to 1980 and have these points actually said. The principles, the methodology, the statistical approach, the display of meta-analysis is 40 years old. And there must be tens of thousands of papers on it. That's exactly the methodology you need to use. Never pool across studies. Always look for differences within studies, calculate variances between studies, wait for the size of the information, which is not just the number of people, but the number of events. Make sure your definitions for your event are reasonably comparable. There's a huge literature, and it's wonderful you're discovering it now. you know, so please, this is not new. Go back and read the literature. And I've been involved in meta-analysis since the 1980s. And the best way to do it is an individual subject approach. You cannot do it for every SAE or AE. You'll just drown in it. And by doing an individual patient approach, you can actually do a time-to-event analysis. So you don't get into the issue, maybe things are happening early in some cases, and other cases it's happening late. You can actually study it. So to the gentlemen and ladies on the podium, please go and read the literature.
Can I add to that?
Sure, go ahead.
Yeah, I think Professor Yusuf, you're right. The tool has been there for a while. And now we have an opportunity to use it smartly with a new set of questions. So for example, in this particular setting, it's not what I call a static meta-analysis. It's a dynamic meta-analysis where when you have data moving ongoings, with this ongoing data, when the evidence still accumulating, that type of decision-making will be different from what I call end of all the trials. Now you want to do a meta-analysis. So that's something, I'm not saying it's not there, but it's in this particular setting, it have a unique, different problem.
So Mary, you seem to have read the literature. You want to?
Right. You're right. It has been around for a long time. And as I mentioned, I learned pretty early in my career a pretty big lesson, and that was a long time ago. So the challenge we're having right now is trying to get good pooling practices into predominant practice. And we still, even though we're years later, we are still struggling with that challenge. So I think there have been conferences, there's a lot of literature, there's a lot of word out there, but somehow it's not getting to everybody who needs to know, because I still think we see a tendency to just pool data, do crude percentages, and look at the data that way. And unfortunately, that's still part of predominant practice. So I think we're all looking for help to try to impact predominant practice. And I know the medical field has similar issues. Usually it takes 15, 20 years to go from a medical innovation to actually have it be part of predominant practice in the doctor's office. So I know other fields have similar problems. But it feels like it's time, right? We say this in the Fuse Adverse Event White Paper. It is time to use these methods, right? It is past time to be using these methods. And so I couldn't agree with you more. They've been around, but we're still struggling with getting it into predominant practice.
I have a couple of minor comments. I'm actually old enough to remember the stuff from the 1980s. But this is more dynamic than a traditional meta-analysis, so that's one. So you've got this cumulative aspect, and you want to know when you're supposed to stop and do something. So that makes it different. It's more like quality control in that sense. And then the other comment I have, which no one seems to address, is meta-analysis. You have to have a certain amount of caution when the events are rare, because so many of the processes that are out there use large sample approximations. And when you talk about big trials, that's fine. I'm sitting at FDA now, working in the rare disease space, and those trials are tiny. So, we need to recognize that it's not one size fits all, and that's one of the cautions I had.
Just an observation. We sort of, and Mark, you probably, in the post-market world, and you're talking about like pharmacoepidemiology, and so many analytic tools to look at real-world data. And the uptake of the newest methods is, just seeming, in comparison, seems to be really fast. Propensity scores and different ways of doing that, and even artificial intelligence, machine learning, and all of those things people seem to be, really wanting to learn really quickly. But then we go back to good pooling practices, and it sounds like, techniques, the best methods from the 80s are still not being used, just beyond education. And just wondering in the back of my mind, like what would it take to increase the adoption of these best methods?
Well, I agree that the methods have been around for a while. The understanding of them has been around for a while. There are some unique aspects, as pointed out, the cumulative dynamic nature. We're also dealing with the rare event situations, which require different statistics. But I think, I think there's a lot of why meta-analysis principles aren't always used is probably similar to why good observational studies principles aren't always used. There's a lot of data out there. It's easy to analyze, and a lot of people don't have the necessary training. In terms of overcoming that, I don't know, I'm not interested, it's not my job to make the whole community, but we hope that our guidance will at least will have some effect in the drug regulation world.
Yeah, great. Next question.
This is a very small historical note. Simpson's paradox was first described in 1899 by Pearson, and then again in 1903 by Yule, and named Simpson's paradox in 1950. So these are pretty old methods.
And I just wanted to add, there's a large literature in what's called cumulative meta-analysis. Tom Chalmers proposed it in the late 1970s. We wrote papers about the problems of cumulative meta-analysis and how you have to use the concept of optimal information size and monitoring boundaries to assess it. You have to think about hypothesis-generating trials, separate that information off, then look at confirmatory data. You call it dynamic, it really is being tested under the concept of cumulative meta-analysis. So I think this process can benefit by good shake-up and thorough delving into the literature. Huge methodological thought has gone into it. And in fact, some of the meta-analysis we did in the 1980s was on safety, on thrombolytic trials. It wasn't just efficacy.
Oh, okay.
Just a quick comment. I mean, definitely agree. I think there's a human nature that always want to the best toy just come up, and then they forget about what exactly near we're hitting.
The shining
so I think the shining part. So I think you are right, and some of those tools has been there for a long time. It's really the fit for purpose is something we forget. We only think about the new thing coming up.
Well, maybe Mark's guidance will have a big shiny
Yeah,
bow
we're
on
waiting for your we're waiting for your guidance.
Next question.
Thanks. I'm Susan Duke, currently at FDA. I haven't been there that long. I have been in industry for quite a long time, though, and have had the opportunity to lead and be involved in industry-wide working groups for quite some time. And my comment today is about culture and how we work together. So actually a colleague of mine who's sitting here, Meng-Chun Lee, she works for a small nonprofit. She's a clinician, and she and I met at DIA last summer. And actually, I'm part of Bill's working group, and we had been looking for safety clinicians to work with. A few months later, Meng-Chun made a little cartoon. And in her cartoon, she has a stick figure in the left. It's a safety clinician at her desk going, hepatotoxicity, adverse events, ECGs, where's the signal? And then there's another panel with a statistician that says, I make perfect methods. I make beautiful graphs. And the panel at the bottom says, the two people are looking at each other saying, we could work together. So I think part of this is each within our own silos, we're developing things and we're figuring things out. And as I have been working on the working group that Bill started, and now that we've added safety clinicians into that group, it's amazing how much we are able to support each other with our respective knowledge and how we can see things. Meng-Chun came to a methods webinar in ASA Biopharm last fall, and she said, I want to know more about those methods. The statisticians thought only statisticians would want to know that. So just giving a few examples of how we can really, as all of us are interested in, all of us have these different perspectives on safety, how we can, if we're working together, we can really get this to where we would like to go. And we don't have to be so frustrated about methods that have maybe been around for decades or centuries not being put into good use.
Great. Yeah, thanks. Any reaction or comments? Okay. Any other questions or comments from the audience? Okay, I think we're done. So let me go ahead and close today's event. I'd ask my panelists to just stay with me here up on stage, but thank you for the presentation and all of your comments. Across today, we definitely learned a lot. The discussion was very productive. We covered a lot of ground and have discussed in depth some of the most major areas of the draft 2015 guidance where there are remaining questions on how to implement these approaches and what the best approaches are for doing IND safety reporting in a way that gets reduced but really meaningful information to the FDA. In the first session, we talked a lot about issues in identifying serious anticipated events. It's not as easy as it seems. There are lots of, we had lots of discussion on how to identify and establish background rates using randomized controlled trial data are best, but some ideas of also when you have to use real-world data, but there are challenges in doing that, and one cannot necessarily mirror the clinical trial population, and so you may not be able to get the background rates that would be the most appropriate for that clinical trial population. But with ongoing work and methods development, that could still be an opportunity as a source of data when it's needed. We also discussed the challenges in using passively reported data versus more structured data, and many remaining questions on the appropriate use of background rates. We talked a lot about Category C type events and when to report and all of those things. So I'm not going to summarize that whole discussion, but moving on to Session 2, we debated at length the issues of unblinding while still maintaining trial integrity and the corresponding roles of the SAC, the DMC, the DSMB+, all of these things, firewalls, questions about the extent to which unblinding only the SAE's cases was brought up and whether or not doing that actually does challenge the integrity of the trial. It was a really important question. I'm not sure we got to the answer of that, but that's a remaining question that we have. And then turning to the most recent, while I was up here, I wasn't able to summarize those last sessions, but you can remember them. Session 3 was on repeating going back to category C type events and what would be the threshold. And I think what we established is that there's no threshold, that it's probably still on a case-by-case basis, but these events should be taken in the totality of evidence. And there was some debate on whether, when reporting to the FDA, that should be characterized in terms of the benefit, or if this is really just about risk, and we need to look at safety and risk on its own for this particular stage. Then we moved on to session 4, which did talk about the methods from back in even before the 80s, it seemed like, but we're still not getting to good pooling practices and opportunities to increase the education, increase the awareness of these good pooling practices. But this all comes back to the overall goal of this and this guidance is not about when to stop a trial or when to change things, but it's more simply about when do we know that there's a risk that's greater than what was expected, and if it is strongly or if it is associated with the particular drug. So with that, I'd like to go ahead and thank everyone for staying with us throughout the day. This was a small group at the beginning, but you stuck with us and you were very, very engaged throughout the conversation, so I thank all of you. I'd also like to thank all of our panelists and presenters. You put forth very on time, but very useful and presentations that were packed full of lots of very useful information. I'd like to especially thank our partners at FDA, Jacqueline Corrigan-Curry, Diane Parowan, and Kahir Elzarad. I didn't look at this ahead of time, I'm sorry. And especially Jacqueline and Lissa for moderating today. That was very helpful to us. Lastly, I'd like to thank the team at Duke, Adam Ayton, Morgan Romine, Catherine Frank, Elizabeth Murphy, and Sarah Saprisi for all of your efforts in putting together. I'd like to thank the folks from the web for tuning in and sticking with us throughout the day. Thank you very much for staying with us. I appreciate your thoughts and comments, and have a great afternoon.